Раз, раз, проверяем звук. Раз, раз, раз.
Здравствуйте.
Утро.
Все слышно отлично.
Раз. So we have many people online. Please indicate in the chat that you can hear me and also you can see the screen. Could you please put plus in the chat or just indicate that you can hear us well? Проверить просто. Okay, it's really nice. So we have many people online. We have blended format, so some people will join us a bit later. So just we have to make few technical comments about the conference. We start in 5 minutes. So right now, some technical information for people online and also here. So I would like to invite Nina. Nina, please. So Nina is the organizer of this conference. Nina, say hello.
Hello, everyone. My name is Nina Arzumanyan, and I'm organizer of the conference. If you have any questions, you can address me always. So be welcome to help.
So Nina deals with all technical issues, including workshops, coffee breaks. So we have actually a coffee break at 1 o'clock. Can we? So we have to switch back to the presentation. Yeah. Okay. So a few words about the today program. I will talk in 5 minutes about our line of research on various manipulations and manipulative strategies that affects our decisions. Later, we will have an invited speaker from State University, like a vision talk on the link between molecular biology, biochemistry, and social decisions. We will have a break. Actually, at the same floor, you can get some snacks at 1 o'clock. Yeah. And after, we have 2 talks by Alexey Belyanin and Ken Chang. So unfortunately, we plan to have a blended format. So 1 talk online, 1 talk offline. By private reasons, one of the speakers today moved online. So we'll have 3 online talks. But we also invite you to the workshop on voxel and surface-based morphometry. So in the same more or less building, it's 5 minutes walk through this sophisticated building, and you will have 2 days workshop. And also we invite everybody for the poster session. It will be in the format of short talks at 6 o'clock. Okay, tomorrow we have more or less the same structure. 2 talks break, 2 talks, and a continuation of the workshop and poster session at the end. The final day will be a bit shorter. We start with invited talk by Nina Kazanina from University of Bristol. We will finish by a workshop on regression analysis by Ksenia Panidi. It will be 2 hours workshop, and also we decided to make a closing of this conference in the form of lab tour. So if you would like to visit our labs, feel free to attend. We will have perhaps a glass of champagne, and we'll show you some labs at the end of the day. So this is the plan. Nina, thank you so much. I will use last few minutes of this talk to present our research center. So we're in the High School of Economics. So this conference is organized by the Institute for Cognitive Neuroscience. Feel free to find all information online. Particularly, International Laboratory of Social Neurobiology was involved, thanks to this laboratory. And we also have to say many thanks to our Center for Cognition and Decision Making that consists of many groups: neuroeconomics group, cognitive control and communication, neurodynamics, theoretical neuroscience, memory, MEG group, and so on and so on. So we can use this occasion to chat with us, also visit our labs, and ask questions about our laboratories. We also have a Center for Bioelectric Interfaces that more or less focuses on brain-computer interfaces and also new methods and neuroimaging methods. So we can find in our institute many technologies from transcranial magnetic stimulation to MEG, FNIRS, and so on. So one workshop is dedicated to MR technique. So we're in this building. If you're for the first time here, you can browse different places here. In the basement, you can find near-infrared spectroscopy laboratory. You can also, on the first floor, find movement tracking, a transcranial magnetic stimulation laboratory. On the second floor, VR lab, eye tracking labs, baby lab. Also, you can find on the third floor, neuropsychology laboratory, brain computer interface laboratory. On the top of the building, social neuroscience neuroeconomics group is located. So feel free to go through different laboratories. We can also introduce you to the laboratories. We will also make a lab tour. Remind you that there is a master program on cognitive neuroscience. So if you're interested, you can also attend this program. There is a nice program of Russian postdocs. Every year we invite a few postdocs with a stipend and also with a place to live in Moscow included. Also, we have postdoctoral fellows for international academics and tenure track position for international academics. So just remember that you have some job opportunities at the university. Okay, so it was a brief introduction to the place, and I think we can switch to our symposium. We have blended format, so some questions will go to chat. So feel free to ask questions in the audience, and also some students and people will enter this room. Don't be disturbed during the talks. So I give this chair to Vladimir Kostanogov. Vladimir is the chairman of our today meeting, and Vladimir guide us through 4 presentations today. Vladimir.
Okay, good morning to everyone. And we begin with the lecture of Dr. Vasily Klucherev with Eliana Monakhova and Yulia Gorodnicheva from our Institute for Cognitive Neurosciences about neuroscience of social influence from euphemisms to deepfakes.
Perfect. Thank you so much. So we also have Yulia and Eliana here. Yes, they will help me to deal with this presentation. If you have some questions, please use chat and we will get your questions later on. You can also ask questions during the talk, okay? So we will talk about our line of research on miscommunication. So for some time, we studied social mechanisms of persuasive communication. So we focused on conformity, on group pressure. So 2 years ago, we summarized our findings in this article in the Encyclopedia for Behavioral Neuroscience, a more or less summary of our views on some general mechanisms of social influence. So here we described our view on the mechanism of conformity, and we decided to move on and study other forms of persuasive communication and focus on miscommunication, misinformation mainly. So we were very much affected by the idea of post-truth era. So this idea emphasizes that currently people focus more on emotionally charged information and on information that fits their beliefs rather than on true facts. And we decided to open a line of research to investigate misinformation. And we are surrounded by misinformation and by some fakes. Some fakes are really beautiful. Now I show you my favorite fake for 1.5 minutes. I think it's beautiful, and I have a question for you. What this has to do with fakes. all fake. Other comments? Okay, that's my favorite fake because it's a famous story, and it was published in this vinyl record. Yeah, and when the person who published newly discovered antique music after his death, we realized that all of this music was created by Andrei Vavilov. Many of these pieces became popular hits, yeah, in classic music. It means it's very difficult sometimes to realize what is fake, what is true. This music is fake, created by a Russian Soviet musician. I really enjoyed this case. He was brave enough to publish a huge list of fake music. And this fake is beautiful. But of course, we are surrounded by dangerous fakes. And we, in our research, focus more on dangerous fakes and miscommunication. So we can perhaps make some definition. Miscommunication is an inaccurate presentation of information. And sometimes, of course, it can use some linguistic manipulations. For example, euphemisms. Yeah? Not all fakes uses euphemism, but they can also use euphemisms. And when we study manipulations and misinformation, it would be interesting to investigate different aspects of misinformation. Of course, the content of misinformation, how do we perceive misinformation, how do we transmit this information, also to study detection technologies and prevention technologies. So in our studies, we more or less addressed all of these stages, and we search for tools to study miscommunication and misinformation. So we will present you 3 studies, and we will offer 3 tools to investigate misinformation. So you can study misinformation, manipulative communication differently. For example, you can focus on the source of misinformation. You can focus on recipients of misinformation, and you can study brains of recipients, of the effective communicators. You can also study brain activity during the processing of persuasive, manipulative messages. Okay? So different groups focus on different stages of manipulative communications. For example, Emily Falk focuses on effective communicators. So for example, in her studies, she showed that brain regions that belongs to the theory of mind network are particularly active and effective communicators. So in our studies, we focus on recipients mainly. So for example, we showed like internal conflicts during persuasive communications, and some of our colleagues, and also we, partly investigated internalization of manipulative information. But in this project, we focus mainly on the content of the messages. Okay, and main majority of these manipulative messages are narratives. So we decided to study narratives, processing of manipulative narratives. And we start with the first research question related to euphemisms. So euphemisms are substitution of the original words that can offend somebody by more neutral words that decrease perceived ethical conflicts of the message. So for example, you're being let go versus you are fired is an example of euphemism that makes this message less emotional. For example, smoking a joint or smoking marijuana, also an example of euphemism that decreases the perceived violation of social norms. So I invite now Yulia to show you our first project that focuses on euphemisms and brain mechanisms of euphemisms.
So thank you for such a bright examples of euphemisms. And the title of my study is Effect of Euphemisms on Moral Judgments, e.g. study. How you might already know, we live in information explosion era. So amount of information we perceive and process has no analogue in history. And of course, it leads to necessity to perform more effective messages to draw more attention of users. Media use a lot of um, manipulative strategies now, but one of the most relevant to present study is media framing. It's when you frame your message with a certain target, and you perform information in a particular way by language means, first of all. That's why we decided to concentrate on euphemisms, because it's very popular now. As you know, it's a kind of indirect word, it's vague expression, it's mild phrase, and still there is no union definition of euphemism among linguists, among other scientists, because it's really depends on context. It's a relative term. One of the brightest examples is to die, to pass away. And moral judgments we evoked with euphemisms, it's when we defining behaviors of or personalities in terms of good and evil. Our main target was to um, evoke N400 correlate, because it's connected with a reaction to norm violation. It might be semantic norm violation or social issues, and it's negative deflection peaking at 400 milliseconds. Our main hypothesis was that the same vignettes representing norm violation with euphemisms are perceived as more acceptable than vignettes that contain no euphemisms. Our neural hypothesis was that ear-peak correlates of norm violations are replicable, like generally for Russian-speaking sample. And second neural hypothesis is that N400 to norm violation is modulated by euphemisms. And it's going to be differences in earpiece at 200, 600 milliseconds for euphemized and non-euphemized conditions. And we hope that low amplitude evident that action perceived as less severe or as normal, and vice versa. We created 120 stimuli. Are they all absolutely the same? Only one word was different. Sorg euphemized norm violations, Sorg 40 euphemized norm violations, 40 non-euphemized norm violations, and 40 fillers. And we included wide range of issues like ethic, business ethic, moral issues, etc. We excluded personal associations like title, names, and we wanted to evoke third-party moral judgments. One of the example is ladies in shop were asked to be quiet shortly after they had a disagreement or they had a fight. For neural data analysis, we use permutation F test with 1D cluster, and for behavioral data analysis, we used one-way ANOVA, where condition was independent variable and participants' evaluations with 5-point scale was dependent variable. While the procedure stimulus was presented word by word, and the last word was different. And shortly after, we asked them to evaluate the norm violation as severe norm violation or less severe, where 0 absolutely right and 5 not right at all. It's resulted in meaningful differences in behavioral data. Non-euphemized conditions were considered by most of participants as more severe, and euphemized as less severe, and control as not severe norm violation at all. in EEG data, we also obtain meaningful differences. The main difference is between non-euphemized and euphemized condition, as you can see. Also, in between, you can see control condition, and I will come back to it later, and I will explain why it can be like that. The highest different wave you also can see for non-euphemized and euphemized experimental condition. Then we defined the region of defined region of interest, and we found meaningful different ways for non-euphemized and euphemized stimuli in 500, 600 milliseconds. It resulted in significant difference of ERP waves around 300 milliseconds and 600 milliseconds, and you can see it's indicated by red color here. So conclusions, we obtained stronger positive ERPs to non-euphemized words around 300 and 600 milliseconds as compared to ERPs to euphemized words and to ERPs to control conditions. So our third hypothesis was partially proved. We haven't obtained any N400 activity, so second hypothesis was not proved at all. And behavioral data analysis concluded in the fact that vignettes representing norm violation with euphemisms are perceived as more acceptable than the same vignettes that contain no euphemisms. So first hypothesis was proved. As you can see, we were hoping to obtain N400, but instead P300 and P600 occurred as sensitive to euphemisms in the context of norm violations. In the literature, P300 and P600 components correlate with different processes like processing of syntactic nonsense and grammatical errors with garden paths. It's phenomenon when revision of a synthesis goes by its long structure. When you have to perform revision of a sentence caused by its long structure, and also its violation of social stereotypes. But more relevant for present study is everyday evaluations of morally unacceptable actions. And the main difference between N400 and P300 and P600 is the fact that N400 is based on knowledge inconsistent stimulus, and P300 and P600 based on moral unacceptable as compared with acceptable sentences. So generally it's based on concepts of good and evil, and our stimulus was more about it. So limitations we had. First of all, euphemisms is a relative term, as I said, and it's really hard to define what is euphemisms and what is not, because it's very depends on context. And sorry for such a sensitive example, but no linguist will consider this as euphemism because it's not even synonyms, but still in terms of context it's used obviously as it. So our further steps is to study relationships between personal traits such as need for cognition and resilience to euphemisms. That's it. Thank you.
If you have some questions, please ask right now. Yeah. We have place for one or two questions.
Yeah, so thank you for the talk. My question is about this P300, P600 result. So basically your interpretation is that euphemisms are more morally acceptable, right, compared to non-euphemisms. So, and Yeah, so my question is, can we separate that from just pleasant or unpleasant? Maybe just euphemisms are more pleasant.
We connected P300 and P600 with norm violation, which is based on moral norm violations, and 400, which is was based on knowledge inconsistent norm violations, when you know rule and rule is broken. And here is we collected like 120 morally unacceptable actions, so it's probably connected with nature of our stimulus.
I would add, this is an important question. So I think right now we cannot disentangle it. So we have to check whether euphemisms are less emotional. So it can be just an index of emotions, but that can be a mechanism of euphemism. So they can be mutually linked. Yeah. So reduce emotion. That's why you react later. We have to speed up a bit. So we'll come back to the questions. I see also Alexey Belyanin has a question online. Alexey, we'll come back to your question later. Yeah.
We actually we're doing the next study with emotional and less emotional words, and I probably will perform any results later because it's slightly different from euphemisms.
Thank you so much. So we continue because we have 3 short projects we would like to present to you on this line of the studies of misinformation. So of course,
yes, sorry, we have also several questions in chat, so I think we can just put them at the end of our presentation, right?
Yes, exactly. Thank you very much.
Okay.
So that's a great idea. We will come back to this at the end. So we have different types of misrepresentation of information. Of course, it can be unintentional. So, for example, some gossips, rumors, and some intentional, from propaganda to lies and fake news. So in our studies, we focused on intentional misrepresentation of information. And next 2 projects focuses on fake news and also on deep fakes. So of course, yeah. So I spent last half of the year traveling between Amsterdam and Moscow. I attended a course on neuroeconomics. I participated in a course on neuroeconomics. Some of you know that Amsterdam Rijksmuseum organized one of the most popular in the history exhibition of Vermeer. I was not able to buy a ticket. All of them were sold, but I started to read about Vermeer more because the event was so attractive. And this piece attracted my attention, a woman reading a letter. And when I realized that it was actually painted by most ingenious forager of art, Han van Meegeren. So he actually faked a lot of Vermeers. And he was so famous at that because he earned, I think, a few, like 30 millions on making fakes of Vermeers. Major museums exhibited his paintings. He owned like more than 50 houses in Holland and 15 country houses. He got problems when one of his fake Vermeer was purchased by Hermann Goering. And it created a case after the Second World War. He was accused of selling national treasures to Nazis. So he was arrested. He faced a court. And actually in the court, he stated that he didn't sell real Vermeers. He faked Vermeers. Nobody believed him. And he had to paint Vermeer in the court, in the prison, to prove that he can do it. Yeah? He managed to do this. Yeah, he painted Vermeer, and he was released from the prison. And of course, we are surrounded by fakes and some kind of deep fakes for hundreds and thousands of years. In the next project, we focused on deep fakes because modern technologies, they of course create much better fakes. For example, a long time ago in the exhibition of Dali, you could chat with Dali. Yeah, it's an old project, but it's still amazing. You can now reconstruct a person. You can deal with the person.
Greetings. Welcome. This technology allows people to imagine for a moment that there is such a thing as immortality. to see DALI alive again.
You can become more.
The whole future of DALI is explained in the brother painting.
So you can communicate with DALI nowadays. And we have decided to study neural processing of deepfakes. And in two next projects will be related to deepfakes and to fake news. But first, about deepfakes, some misinformation created with use of artificial intelligence. And I give the stage to Eliana. So Eliana tried to make a first study in our group of deepfakes.
Hello, everyone. I'm very happy to see you on the conference, guys who are offline and online as well. And basically, I would like to present you my topic, which is neurocognitive processing of attitude-consistent and attitude-inconsistent deepfakes and 400 study. So today we will discuss the theoretical background of my research. So I will outline the term of deepfake and the N400 component again. Likewise, we will discuss aims and hypotheses. We will look into methodology and experimental procedure of the study, and of course, turn to results which we got and limitations of the research. So as Vasily Andreevich already mentioned, we were dealing with deepfakes, and practically, deepfake is a video or sound recording which can replace someone's face or voice with that of someone else in a very realistic way. So such format of disinformation distribution is effective because it's very close to the reality. So like practically, as an example with Dali, you can get absolutely new person, which is unreal, but very realistic. And of course, due to the effectiveness of such fake type, the level of trust of it is very high, and it's influenced usually as well by congruence to viewers' internal attitudes, as previous studies suggest. But of course, there are some internal characteristics which may help to counter with such fake formats. And it is known that people who are more reflective, they are usually less likely to fall for the untruthful content. Likewise to the deepfakes, and it happens in the case of even if the information is inconsistent to people's views. So as my colleague Julia has already suggested, we are likewise dealing with the N400 component. I would like to remind you that this is a component of event-related brain potential. It shows a negativity peaking at about 400 milliseconds after a stimulus onset. So the stimulus can be different. It can be a word, it can be a audio message, likewise some video fragments, so anything which may be perceived by the person. And what is interesting for my research, I tried to deal with the continuous speech. So we will discuss deepfake speeches because previous studies show that such effect may be found not only in written sentences, but likewise in spoken words, and usually in the sentence final position. So now let's turn to the aims and hypotheses of my study. We firstly decided to evaluate the degree of trust to attitude-consistent and attitude-inconsistent deepfakes. They were devoted to the topic of COVID-19 vaccination in Russia. And also, as for the neurophysiological aim, we decided to observe earpiece, VN400 component particularly, at the end of the sentences, which would be uttered by deepfakes, violating participants' internal attitudes and expectations. As for the hypothesis, again, they are divided into behavioral and neurophysiological one. The behavioral says that people tend to trust more in audio messages, in our case, deepfakes, that are conveying their internal attitudes. And as for the neurophysiological hypothesis, the amplitude of the N400 component will reflect stronger negativity in the case of incongruence to participants' attitudes and their expectations connected with public opinion of the speaker. We will discuss this a bit further. So for our experiment, we invited 60 participants. 8 of them were excluded due to bad EEG data. They were divided into 2 experimental groups, pro-vaccinators and anti-vaccinators from COVID-19 virus. As we dealt with EEG recording, they had to have no history of neurological impairments. Likewise, they were obtaining higher education. And what is important, then they had to be acquainted with the public opinion of the speaker. Why it was important? Because we had 2 different speakers. who were translating audio messages which were opposite to their public opinion. So basically, we had one speaker who was a doctor and another one who was a COVID dissident. And in our deepfakes, they changed roles and translated absolutely opposite opinion about vaccination. And of course, all participants should have been unaware about the fact that this experiment includes deepfakes. So they got to know it only in the end of the experiment. So likewise, in addition to our audio deepfakes, we had the control N400 paradigm. So it consisted of 40 congruent and 14 congruent sentences. But firstly, I will show you the example of one of the part of our doctor's deepfake speech. So it consisted on three different types of sentences. So one was incongruent, which had the word in the sentence ending, which was incongruent to his public opinion. Concerning filler phrase, it had to be something to hold on the logic of the narrative so that people would not guess that we're trying to fool them. And the congruent sentence was the sentence with almost the same word as the incongruent had, but it was consistent to the public opinion of the speaker. So, as I've already mentioned, we had an EEG recording of 32 electrodes, and the timestamp we used was congruent and incongruent words onset. So, if we had like the word beginning, we were focused on it and on the word duration as well. Next, we likewise added some questionnaires, which were cognitive reflection test, need for cognition scale, and conformity scale. And as well, we had several statements which were assessed by the participants after deepfakes listening. They were connected with the trust, so we were asking how much they do trust our deepfakes. And as for the statistical analysis, we had two different types of it. The first one was multifactor ANOVA in 2 trials, and likewise, we had permutation F-test with 1D cluster level. So we were inviting participants, they passed screening questionnaires, they came to our lab, we put on EEG, and they listened to our 2 deepfakes. After that, they had 5 different statements of trust. They had to evaluate them from 1 to 7. After this, they passed control N400 paradigm, reading different sentences from the screen. And in the end, they were fulfilling questionnaires and personality tests and passed debriefing procedure. So as for our N400 classical paradigm results, you can see that we had different types of sentences, as I've already mentioned. So the congruent one was, for example, I drink coffee with milk and sugar, so logical ending, and the incongruent had jacket in the end. So this semantic evaluation, this semantic incongruence, it provokes this N400 component elicitation. So basically, in this picture, you can see that the orange line stands for the incongruent sentences ending and the blue one for the congruent. And we can definitely see that there is a high difference in the in such words perception. So we elicited this reaction. And we tried to do the same for the main part of our experiment. So as for our doctor deepfake, we managed to see something like to the N400 reaction on 600 milliseconds after the stimulus onset, and it was observed in the anti-vaccination group. We haven't seen such reaction in pro-vaccination group for the speaker, and we tested the reaction for the second speaker, which was COVID dissident. Likewise, as you can see, none of such reaction was detected. And we decided to see whether this reaction is really significant. I don't know why it's not red, but actually here, the reaction between congruent and incongruent sentences, it was significant enough. So basically, our people from anti-vaccination group reacted to our doctor's deepfake in such a way. The only thing is that this reaction was postponed for 200 milliseconds, and it was on incongruent words. So basically, when the doctor was saying that vaccination is bad, our anti-vaccination group was reacting in such negative way. Next, we decided to conduct ANOVA, and the first trial was with a dependent variable of the N400 like difference wave amplitude. So what is this difference wave amplitude? We were taking incongruent wave and congruent wave, we're making the like one minus another. And we understood that there is no significant factors influencing its difference. Next, we decided to see whether the same factors were influencing the level of trust to our deepfakes. And we managed to find that two factors, such as speaker and the score in need for cognition scale, were highly influencing the trust to deepfakes. And also on the picture, you can see that our doctor's deepfake was trusted much more than our COVID dissident deepfake. And it was likewise proved here. So you can see the trust score, the mean score for each group for each deepfake speaker. And with also by Tukey's test, we managed to find that anti-vaccination group tended to trust our doctor's deepfake significantly more than our COVID dissident. Okay, so what is the summary of all our experiments? Basically, both of our hypotheses, behavioral and neurophysiological one, were partly confirmed. Why the first one was partly confirmed is because anti-vaccinators really trusted doctors' deepfake more than COVID dissidents' deepfake. And to remind you, our doctor's deepfake was saying that vaccination is bad. So, like, opposite view to his public opinion. And likewise, we found out that the level of trust was moderated by two factors. It was speaker and need for cognition, not internal attitudes. So this is the part which was not confirmed because the hypothesis suggested that internal attitudes moderate trust. And for our neurophysiological hypothesis, we found that anti-vaccinators had higher negative evoked response to incongruent words of our doctor's deepfake speaker. So what we think about this? We speculate that for our doctor's deepfake, pro-vaccinators trusted him unconditionally because doctor has a high authority towards this group, and that's why they were not critical to semantic inconsistency. So basically, whatever doctor says, they still believe him, and that's why we haven't detected this N400 effect, because it usually appears when we have semantic anomalies and likewise expectation anomalies. As for anti-vaccination group, we decided that they trusted our doctor conditionally. So it was connected with the fact that his authority was much lower in this group, and they did trust the particular message, but they were not respecting this speaker like fully. And that's why they were critical to semantic inconsistencies, and this N400 effect presented in one of the conditions. As for our COVID dissident deepfake, both groups, they did not trust it, and the trust score we counted also shows it, because this is an influencer with a low speaker's authority. And that's why no semantic inconsistencies appeared, and we haven't seen this N400 effect, basically. So let's move to the last slide about limitations. We guess that one of the main limitations we have is that this doctor who was our speaker, he could be perceived more authoritative and deserve more trust just because he is more, he has more competence in medical issues, because he's connected with the sphere and he knows better than the COVID dissident, absolutely from another sphere. And likewise, some participants recognized that these materials we were presenting to them were fakes, but they were fervently deleted from the analysis. So all the data we have is clear enough for making such study.
Okay, we have a space for one question in the audience. Yes, please.
Hi, my name is Alex. Excellent presentation. But the question may be, what is your opinion when you look at the brain? Does it has a built-in biological mechanism that can help to differentiate between information and misinformation if trained enough, or the opposite, that the brain can always be tricked no matter how well you try to convey the truth?
Yes, thank you for the question. I think that I can answer in such a way that there are a lot of factors which can influence the level of trust. So basically, even if the person is very well trained, so he has like high internal characteristics of need for cognition, he's very reflective, he has high analytical thinking, Still, he can be tricked, of course, by the fake, depending on its quality and type of representation, I guess. As for the brain, in my particular research, we were trying to detect this ERP, the particular ERP, the N400. And the thing is that when we try to look at these topographic maps, the ones which I show on the slide, the activity of the brain was actually situated in different areas. And I can answer like it's very hard to detect which part of the brain really responds for the information and misinformation perceiving. So we were practically focused on the concrete ERP. So I can answer about this. Like we detected in the occipital part of the brain for one group, and the reaction for another groups in different conditions is under question.
Perhaps we cannot answer all questions using one experiment, but this is a great question, yeah? Also for discussion. Thank you very much. Since we have also a lot of questions online, I will skip the third study and I will move to the end of my talk. Just give me a sec. Can you, it's, yeah. So because we have many questions, I think we can focus on the discussion. So we just started 3 years ago this new line of research, and as I mentioned, there are different aspects of misinformation. Yeah, so we try to study how does brain process the context of the messages. So, for example, we focus on euphemisms in Julia's studies. So we also focus on deepfakes and processing of the information using N400 technology. We're also in many projects which actually study intention to transmit this information. We haven't analyzed this data. We perhaps can deal with our computer scientists who investigate some techniques, like, for example, Vasily Gromov, who makes techniques to detect bots based on the linguistic aspects of their communication. And also Alexandra Kuznetsova, I don't know, does she make a presentation in this conference? Perhaps not. She studies various techniques how to correct for fakes, for example, debunking technology. So what I suggest to do, actually, if you're interested in information, please contact us. We will be very happy if you're a student or a senior researcher. We're real open to collaborate. But now we have space for some questions. Zarina, can you, because we don't see the chat, can you name some of the
yes,
questions?
sure. Sure. We have questions for Yulia, I suppose, from Alexey Belyanin. So probably, so maybe Alexey would like to ask them by himself. If not, I can do that. So, okay, I will read it. So, the first question: how exactly do you validate strengths of moral or social norm which is being violated?
There is if definitions of good and evil are included, it's mostly relative to moral norm violations. It's
if I may, sorry, just to clarify the question, right? I am playing here the role of devil's advocate, right? I'm placing myself in a position of a referee who is trying to destroy the result. Okay, claim that what we observe is that your norms are not really that strong, and people's interaction between the strength of the norm and emotional reaction to the stimulus is different for different people, right? So you don't really have a stable stimulus shown to everyone, and that's why the result is shaken by these interactions, and you don't control for them. So what would you say to that? Thank you.
Thank you for the question. I would agree generally. with it, but can I also involve the results of my next study a little? Because there is uh, we're looking for difference in reaction to emotional and less emotional words, and we did pretesting for emotional and less emotional synonyms, and reaction was more or less homogeneous. So we can think that partially, at least, reaction to the same emotional and less emotional words are at least partially the same for different people.
Great question. So perhaps at the level of publication, Yulia has to take into account this comment, yeah, either by additional test or also Yulia has a lot of behavioral measures. Perhaps she can study the distribution of responses. So we have space for at least one, I think, question for one. Yeah, Zarina, can you help us? Just read it.
So that's it actually
Okay,
from the
perfect.
chat. Probably, yeah, if
Since
anyone
we're
want to ask.
a bit late in time, so I suggest that we stop here and we can answer questions during the break. And thank you so much, Yulia, Ilyana, thank you very much. And I give this microphone to our chair of the day, to Vladimir Kazanov.
The next talk will be given by Dr. Raul Gainetdinov, the director of Institute of Translational Biomedicine, St. Petersburg University, about emerging pharmacology of trace amine-associated receptors, emotional and social dimensions. Please.
Just a second. Good morning, all colleagues. I'm very happy to present here my talk. And let me open my presentation. Okay, that's it. So can you see me?
Yes.
So you see the presentation and you can hear me, yes?
Yes, please.
Okay. So should I put myself on screen or it's okay like that?
It's okay.
It's okay, yeah? So today I would like, first of all, I would like to thank Anna and Vasily for inviting me to this interesting meeting, and I'm happy to present my data and thoughts about this emerging field. So you all know well about dopamine, serotonin, and norepinephrine and their role in cognition, decision-making, emotions. And is there something else? Is there something else which could also influence what other monoaminergic neurotransmitters can influence this cognitive processes? And in fact, yes. And these are so-called trace amines. Trace amines originally were known characterized as beta-phenylethylamine, tyramine, tryptamine, octopamine, but now it's clear that there are many more of them. They were found in mammalian tissue at low concentrations, usually about several hundred times lower than dopamine and serotonin, norepinephrine. And they're small lipophilic substances, monoamines, structurally and functionally close to classical monoamines, such as dopamine, serotonin. In fact, they were discovered more than 140 years ago, about 30, 40 years before dopamine and serotonin and norepinephrine were described. But the function was still relatively little known. They called trace amines because they're low concentration in the brain. They turned as a false neurotransmitters in mammals because it believed that they just indirectly modulate dopamine and serotonin. But in invertebrates, for example, tyramine and octopamine are major neurotransmitters. Octopamine originates from the word octopus, by the way. They present in food, and I will focus on that later. And in fact, they're just products of decarboxylation of amino acids. So what is the process of decarboxylation? Is amino acid. If you cut acid part, you left with amines. That's our trace amines. So it's part of amino acid. So
I'm sorry, Dr. Gainedinov, could you make a full screen, please?
It's not full screen, yeah?
No.
Okay. Yeah. So trace amines are importance of trace amines in neuroscience, neuropharmacology is difficult to overestimate. For example, just because of this fact. So beta-phenylethylamine, the best studied trace amine, gave name to such important molecules as dopamine and amphetamine. Dopamine, in fact, is just 3,4-dihydroxyphenylethylamine. So it's just these two OH parts differentiate from beta-phenylethylamine. And amphetamine is just alpha-methyl-phenylethylamine. Because of this alpha-methyl group, beta-phenylethylamine is not being metabolized by enzyme named monoamine oxidase. Because beta-phenylethylamine, in fact, is endogenous amphetamine, but which is quite quickly being metabolized by monoamine oxidase. So some people call beta-phenylethylamine as endogenous amphetamine, and some people name amphetamine as unmetabolizable beta-phenylethylamine. And effects, infection, neurochemical effects are pretty much the same. So they're pumping out dopamine out, and the concentration of dopamine goes up after beta-phenylethylamine or amphetamine. But in case of beta-phenylethylamine, is very fast for like 20 minutes only. Interestingly, the story of trace amines have some relevance for St. Petersburg. In fact, beta-phenylethylamine was first isolated by famous Polish, at this time he was a Russian citizen, Marcel Lenensky. He found it is in 1876 as a product of decarboxylation of phenylalanine from rotten egg first, then egg white, then from gelatin, then from meat. So he found it and described it first when he was working in Switzerland, in Bern. And then, by invitation of Ivan Pavlov, he joined Institute of Experimental Medicine in St. Petersburg, where he was working for about 10 years and unfortunately passed away there. And in this photo, you can see in white is Ivan Pavlov, but I cannot recognize who is here, Marcel Lenensky, because they all look the same, bearded, you know. Today, major Institute of Neuroscience in Warsaw, in Poland, named after Marcel Lenensky. So, as I mentioned, trace amines are a result of decarboxylation of amino acids, and the same enzymes actually used by dopamine and serotonin, L-aromatic amino acid decarboxylase, and metabolized by monoamine oxidase. Again, the same enzymes which used by classical monoamines. As I mentioned, concentration is several hundred times lower than dopamine and serotonin. But because of they cannot be stored in the vesicles, that's why probably concentration is low. Interestingly, the rate of synthesis is comparable to dopamine. And release is insensitive to depolarization, so there are a lot of miracles how it's really functioning. This is first I was talking about endogenous trace amines. Now, trace amines also can be produced in various foods because bacteria have their own decarboxylases, and different types of bacteria has their different types of decarboxylases, and that's why every foodstuff which has bacterial fermentation and process, they have a lot of trace amines. So cheese, wine, cured meat. As I mentioned, in invertebrates, in seafood, they are endogenously produced at significant amounts. So all this tasty stuff actually moved us with my wife to Italy, really to taste these trace amines, to understand. You know, in all time, pharmacologists, what they study, they tried it to eat, you know, to try to taste it. So that's why we were tasting with Tatiana, my wife, for more than 10 years. We were tasting this trace amines with great pleasure, by the way. Chemistry of chocolate. The trace amines have a lot of in chocolate, and actually a lot of speculations were that about trace amines in chocolate, but it's very difficult to say. The chocolate has a lot of components, including trace amines. Beer. Beer is a product of fermentation, and when today evening you will try to get your pint of beer, just think about this fact. Look how many trace amines are there: phenylethylamine, tyramine, tryptamine, putrescine, cadaverine, spermidine, agmatin. They're all considered today as trace amines. So, what the function, where they're reaching the brain at significant concentration is a big question, but you surely will get it today. Soybeans. Soy contains a high concentration of phenylethylamine, and if it has phenylethylamine, it has better phenylalanine. It has better high concentration of phenylethylamine. Actually, the topic of trace amine was quite famous in the 70s when it was found that some foods containing high concentration of tyramine actually can cause so-called cheese reactions. The fact is, in 70s, there were quite popular antidepressants were monoamine oxidase inhibitors. And when the people consumed high concentration of this food with tyramine, plus MAO inhibitors, they had so-called cheese reaction because trace amines in the periphery were releasing norepinephrine in amphetamine-like way. High concentration of norepinephrine caused vasoconstriction, acute hypertension, severe troubling headache, and even sometimes death was happening. So that's why, as I mentioned, they work like amphetamine, pumping out classical monoamines, in this case norepinephrine, outside of neuron and increasing concentration of extracellular concentration of classical monoamines. That's why it was actually a warning when people were using these monoamine oxidase inhibitors. They were warned not to eat cheese, soy sauce, teriyaki sauce, alcoholic beverage, fermented tofu, kimchi, salami, smoked processed meats, sauerkraut. So it was warning, official warning. So another source of trace amines are microbiota. In the gut, we have a significant amount of bacteria, which is chopping amino acids and creating these trace amines. And what is the role in physiology, pathophysiology? A lot of speculations today about microbiota gut-brain interaction, how it influences our brain. And this is absolutely unexplored topic right now. I'm expecting to see huge papers sometime soon in Nature and Science on this topic. So summarizing that, our major sources of trace amines, it's endogenous production, food, gut, and don't forget skin microbiota. So millions and billions of bacteria and fungi lives on our skin and producing these trace amines. And another source is pathological production by bacteria during infection. Every infection creates trace amines, so think about that. And that's all was known about trace amines until in 2001, this paper appeared by a company, Synaptic Corporation, actually was looking for new ligands of receptors named G protein coupled receptors. I'll explain you what these receptors are. I was at the moment at Duke working, I will tell you, with Mark Caron and Bob Lefkowitz, and a company was looking actually for new serotonin receptors. It's a company was looking, they're finalizing companies. They were looking for new neurotransmitters affecting new unknown receptors. So, and we were reviewers of this paper. Of course, we were very excited. And when we were invited to write a commentary, following the trace of elusive amines, and for 20-something years now, I'm following this path. And the longer I'm going, following this path, the more exciting it becomes. And I will try to share our recent data on that. And interesting organization of these receptors. They're all located on a single chromosome, one after another, the same way like olfactory receptors are, because for dopamine receptors, for example, one receptor on second chromosome, another one on sixth, and so on. So it's distributed over genome, but here is one after another. And these so-called G protein coupled receptors or 7 transmember receptors, 7 TMR as Bob named, where this topic was created by such wonderful scientists as Bob Lefkowitz and Mark Caron. And I was fortunate to work with them for 12 years. So what GPCRs are? This is a machinery to transfer external signal inside of the cell. Pretty much I can explain you like that. And what are the neurotransmitters, hormones, peptides, glycoproteins, lipids, ions, tastants, odorants, photons of light. Rhodopsin is prototype GPCR, just how we see. And at the moment, Bob and Mark were working for that like from 1971, and they discovered the first one, beta-2 adrenoreceptors, receptors activated by adrenaline or epinephrine. And they were, when they were writing one of the first papers on that, they claimed that there would be many more similar types of GPCRs, but they couldn't even imagine how many they are, in fact. 826 GPCRs are known today. It's like dopamine, serotonin, cannabinoid, epinephrine, norepinephrine, and you can go and so on. So muscarinic receptors, histamine receptors, you can go and go and go. So it's not surprising that 30–40% of current pharmacology based on targeting of GPCRs, either activating them or blocking them. And the role is in the brain and all over the body. It's huge, as you imagine. So in fact, there are 350 somatic GPCRs and 150 additional orphan, so-called orphan, that we don't know the function of them. We don't know ligands what activate them. Maybe now less than that because time is changing. Some people are finding ligands. And the rest of them are olfactory receptors. So that's a genealogic tree of GPCRs. And you can see I circled here. That's the so-called receptors for trace amines, TARs, trace amine associated receptors. So just around them, dopamine receptor, adrenergic receptor, serotonin receptor, histamine receptors. And this is the last group of monoaminergic receptors not characterized so far. And I expect, and just imagine how huge pharmacology is around the receptors and roles, physiological roles, and whatever you can describe with all these receptors. And trace amine associated receptors as the last group is the last group of receptors which can, in my opinion, create absolutely new pharmacology and give us a lot of ideas how brain is functioning in general, not only brain. So originally, if you take a look at the second column, that's what their old name, TA1, GPR58, 57, TA2, TA4, and that's what old names. Now they renamed like by order of appearance of chromosome TAR1, TAR2, TAR3, TAR4, 5, and so on, less than 1 TAR9. So humans have 9 genes, but 3 of them unfunctional. So, and I now created new classification. In fact, in the left, you see they are so important that in Estonia, they have even beer named TAR, as I found recently. So that's, I created new beer classification. You can see the functional one, TAR1, TAR2, TAR5, TAR6, TAR8, and TAR9. That's the only functional genes what we have in our body, and of course we are focused only in functional ones. The other genes are so-called pseudogenes, so they don't create a protein or functional protein. In fact, TARs were probably known 3,000 years ago when this city was developed in Italy, in Sicily, Italy. Taormines, they just made some mistakes, this beautiful city. We were visiting it a couple of times. They just a couple of mistakes in the title, so it should be tyramine. But because probably Italians have the best concentration of trace amines in the world in their food. But I was really impressed when I came there and said, "Oh, look at that." So, as I mentioned, trace amines are products of decarboxylation of amino acids. And when we were writing this review with my colleagues from Switzerland, from Hoffman-La Roche and Canada, Mark Berry and Marius, Henner. We came to this table, which I name Mendeleev table of trace amines. So we have 20 amino acids. So theoretically, for every amino acid, if you cut acid part, it should be amine. And in fact, if you look at the tar ligand, many of them already described as tar ligands. For example, from ornithine, you can get putrescine, and this tar 13 agonists in some species. Okay, there's a different number of tars in different species, and in some species already were shown. Phenylethylamine, beta phenylethylamine. Of course, it's all the tar agonist. Tryptophan, tryptamine, tar agonist. Tyrosine, tyramine, tar agonist. Formalin is a butylamine, not described yet, but I'm hoping to see it soon. Arginine, agmatin, tar ligand. Histamine, histidine, histamine. Even histamine is tar ligand as well, and so on. You can go and lysine, cadaverine. And for example, putrescine and cadaverine, that is a odor of rotten tissue. It's a dead body smell. That's a major component of rotten tissue. The best studied is TAR1, and it's already the first was created a knockout and was described as a model of schizophrenia, and we were investigating it together with Hoffman-La Roche, first with Lundberg, then with Hoffman-La Roche, and 50 papers later, I just can share so that it's already drugs being developed, and it's shown that it's expressed in the brain, in limbic predominantly brain areas, and it has potential antipsychotic-like, the TAR1 agonist, potential antipsychotic-like activity, potential antidepressant-like activity, potential procognitive activity, and even influence wakefulness in rats, and in humans too now, it's already shown. Potential of TAR1 agonist in addiction. It was in animals, experimental animals. It was reducing intake of amphetamine, methamphetamine, nicotine, alcohol, in morphine even now, and food in binge eating protocol as well. It can influence diabetes, obesity potentially. It's all experimental data. Very interestingly, it's expressed in all leukocytes, and it can influence immune system and found in cancers also. Quite interesting papers now appearing, and we also contributing to these papers, to this literature right now. So summarizing, TAR1 agonists have a potential in the treatment of schizophrenia, ADHD, bipolar, stress-related disorder, depression, addiction, diabetes, obesity. It's just, I believe this list will be increased as the more we'll study that. Now we're investigating, for example, post-traumatic stress disorder. The most advanced thing happened with TAR1 agonists developed by two companies, Roche and Sunovion, now in second phase, Roche in second phase, and Sunovion and third phase. And the first data from second phase of ulateron showed that it's actually so important that it's FDA has awarded designation, breakthrough therapy designation for the treatment of schizophrenia. What it means? It means that it can speed up development of the drug by 2-3 years. Interesting names they put for the two compounds. Yes, you can see, I'm really surprised, but I was a major expert involved in both companies involved for development of these drugs. So it's quite interesting. So I'll skip the data, just say that ulateron in this phase 2 study showed very nice results on both positive and negative symptoms. And most importantly, it had very little side effect characteristic, for example, for haloperidol. If you know, haloperidol create so-called extrapyramidal side reaction. It worsens cognition and has metabolic effects, all of these other antipsychotics. And ulaterone has very little effect on that. And ralmitarone right now on second phase, as I mentioned, but we don't know yet the results of these studies. So why suddenly, this is very complicated slide. Just look in the center. You see in the center TAR1 and D2 are together. It's D2 dopamine receptor. So TAR1 is, as we believe, TAR1, how it works. TAR1 makes a direct physical connection to D2 receptor, and it's so-called heterodimerization. That's a quite popular concept now in GPCR field, that the receptors can work together. And when these receptors together, now usually all antipsychotics, currently used antipsychotics, they just block D2 dopamine receptor. It's like hammer, okay? It's very potent effect, just blocking D2 dopamine receptor. Here you have activation of TAR1, and by activating TAR1, you change the properties of D2. So it's just, instead of hammering, you just fine-tuning D2 dopamine receptors. That's why effects are mild, and that's why we believe they have less side effects. Probably it's enough to modulate psychotic reactions, but not strong enough to create side effects. And here I would like to show you a video which was created by Sunolian about how TAR1 works. And I was consulted on this video. Okay. So we continue to work on that, and I will stop now about TAR1 because other receptors are very interesting as well. So there are a lot of, you can see some titles of what we're doing. We're looking for some new compounds. We're trying to find function in periphery in the brain, and so the topic is developing very well. So what about other receptors? And the best studied is TAR5, the next best studied. It was actually discovered before TARs were known as orphan receptors, and it was described that named putative PNR, putative neurotransmitter, and was shown that it's expressed in skeletal muscle and in brain, in amygdala particularly. But the data were so weak that it was not impressive. Nobody believed it actually. So, and even more doubt about other receptors came from the point of view of pharmacologists in 2006, when now my friend Stefan Lieberles and Nobel Prize winner Linda Buck received with Richard Axel a Nobel Prize for discovery of function, how olfactory receptor function. So for this discovery. So she wrote Nature suddenly appears second class of chemosensory receptor in olfactory epithelium, which was claimed that all other TARs, except TAR1, are located in olfactory epithelium, and they sense innate odors, decomposed tissue, predators, sexually relevant odors. And it's okay. We kind of were very excited about that. But what I was really disappointed, because they claim that it's not found in the brain. So how they did it? They just took whole brain, homogenized it, and looked for mRNA. But you can probably dilute it if it's expressed only in certain nuclei. So, and I was very unhappy with that. And all my rest of career, I'm trying to prove that this paper is wrong. And in fact, the TARs are expressed in the brain because I already had some data. I was working already on this TAR5 receptor. And I'm trying to prove now that they mistakenly excluded the role of TARs in the brain, not only in the brain. So in fact, we can see on the left, so this is olfactory epithelium, and only when it says olfactory epithelium, they claim that receptors expressed here, then they, from neurons projecting from glomeruli. You see glomeruli, and that's it, and that stops. And then another neural, mitral cells, and some other cells are projecting to the brain. So that was all. It's only expressed on the olfactory system. That's what they claim. And as I mentioned, decomposed tissue. So it's clear. It's decarboxylation bacteria, decarboxylate amino acids. This is this smell of cadaver in putrescence and so on. That's clear. And sexually relevant odors, pheromones. How it's happening? On skin, we have a lot of bacteria. Depending of bacteria, we have certain composition of trace amines, which can affect this receptor. So trace amines, olfactory trace amines, aromatic trace amines, they're different in every body because of bacteria. Bacteria, the composition of bacteria depends on type of immune system. We have different types of immune system based on HLA, different types of HLA, histocompatibility complex. So human histocompatibility complex, HLA. Depending on that, we have different types of and evolutionary, male and female, looking for somebody which has different type of immune system, that their kids will be having more wide type of immune system. So that's why that's not my hypothesis. It was published before trace amines were found. So by smell, we can predict type of immune system. That's it. Predator's odor. How antelope knows where tiger was, or mouse know where cat is? Very simple. My friend Steve Lieberles went to zoo in Harvard and collected urine from 400 animals. And predators were different from okay. Predators were having higher concentration of beta-phenylethylamine. Why they have higher concentration of beta-phenylethylamine in urine? Because they consume meat, and meat is rich in phenylalanine. Very simple. position. So, and in fact, now I cannot say something about this book by Patrick Zushkin and now movie Perfume. You remember the story that one perfume maker was collecting bodies of dead, killing some girls and collecting them and collecting smell of the aroma of them. And in fact, what he was collecting, these are trace amines, and he was hoping to develop the most amazing perfume ever. So I cannot say it. I mean, Zivskin wrote this book like 20, 30 years before anything was known about TARS, about what we know. It was kind of predictor. I mean, I cannot imagine how he wrote this book, but it's in fact this is a prehistory of trace amines. And interestingly, very interestingly, this trimethylamine was found to be the best agonist of TAR5. Trimethylamine, what it is, it has rotten fish odor, and it created. It's not actually a product of amino acids. It's from choline, appears, but still considered as a trace amine because it's activated potently at TAR5. Again, cannot say something about perfume. If you read the book or watch the movie, this guy when he was born, he was put by her mother was selling a fish on the market, and he was put in a basket with rotten fish. And first day spent life there, so he really was inhaling trimethylamine. He should be. That's the most amazing thing how it was happening. Interestingly, our German colleagues show that the best blocker of that is a compound called timberol, which has amber woody odor. Very nice. It's an artificial compound, and it's very interestingly. So, okay, you have something sensing rotten odor and blocked by something which is have amber woody odor. What it means? Now, just remember funerals. And in Russia, we used to put, there are some yolka called yolka, some wood, piney wood. We have to usually put there piney wood. Just remember, church, ladon, mirror, and ladon and mirror is product of хвойные деревья тоже. Piney woods, it's also piney woods, okay? And they all contain high concentration of timberol-like compounds called terpenes. They all full of terpenes. And we came to idea, why not to try now several terpenes? And we tried in our test system several terpenes and ladon. And we found 3 types of ladon. I'm telling you, it's just by secret. It's not published yet. It's just unpublished observations. We tried if ladon and several terpenes block trimethylamine effect in vitro in our test system. And they worked. They worked, and it's kind of now opening new field in pharmacology, because there are about 30,000 terpenes. So now we are planning to test many terpenes. We tried like 5, 6 of them only. We're now planning to get a collection of these terpenes. So pretty much terpenes are found in any pine tree, in any citrus, I mean, lemon or whatever, orange, many different trees, many plants. And we now planning to systematically to test all of them, no, maybe not all of them, but part of them for effect on trace amine associated receptors, particularly TAR5, and hoping that it will create some new interesting ideas for pharmacology. And why we believe that it's important for pharmacology. In fact, we have TAR5 knockout mice. But it's not only knockout. We have some insertion of marker. We can see where this receptor should be found. As I mentioned, our American colleagues from Harvard, they showed it that it's only on first neuron. First neuron getting to glomeruli, and that's it. And you can see it here in blue. It going to glomeruli, and they say it stopped here. It doesn't go inside of the brain. But when we looked at the behavior of mice, they show like anxiolytic-like behavior, antidepressant-like behavior. They have alterations in serotonin levels in several brain areas. They have other manifestations, which is suggest that it does something in the brain. Come on, it cannot be just olfactory receptor. And make the long story short, when we looked at the expression of this receptor, we found it in limbic brain areas where projection from olfactory system goes. It's the oldest emotional brain, and it gets olfactory information. I will not go in detail that you can read it in the paper. But in fact, how it looks now, that we see like even projections, you can see on the top how it goes to the brain. And so in fact, this brand new, we believe is brand new neurotransmitter system, transforming innate olfactory information into emotions. And we find it in every brain areas, what you can see here. And we believe that's new untapped source for pharmacology now. So we can now play directly with emotional brain. So so that's a conclusion. You can read it quickly. It's the same thing what I say. So let's go further. Another thing what we found, that these receptors are found around ventricles in the brain and in neurogenic zones. So just to think about that, they sense something rotten, something decomposed tissue, yeah, and they round ventricles. Maybe they sense what's happening in the brain through CSF. You know, maybe something wrong happening. They can detect it now. Interestingly, and then they exactly the place where at least SVZ subventricular zone where neurogenesis occurs. Another observation we found that dopaminergic system is increased. A number of dopamine neurons is increased. So, and dopamine concentration is increased. So, and maybe we thought that neurogenesis could be affected in these animals. And in fact, two zones are known for neurogenesis in adult neurogenesis in brain. It's a so-called subventricular zone or dental gyrus or subgranular zone of hippocampus. It's the two major brain areas where adult neurogenesis occurs, and it's a lot of papers about that, thousands and thousands. And in animals, it's proved very, very well. And in fact, now many people suggest that neurogenesis may occur in some other places as well. And some people suggest that even dopamine neurons can appear, can be restored somewhat. And what I like with this neurogenesis story, there is a hypothesis that Parkinson or Alzheimer occurs when the number of newly born neurons less than constantly dying neurons. And this balance is changing. That's what brings to neurodegenerative process. Some people think about that, like that. And neurogenesis can be affected by factory learning. Positive modulators, environmental stimuli, learning, cognitive activity, even physical activity affects neurogenesis. Estrogens, antidepressants. This is one of the major theory in depression now, in action of antidepressant, that antidepressants just increase neurogenesis in the hippocampus, in dental gyrus. Negative modulators, stress, aging, glucocorticoids. So, and that's now very exciting topic for research, and I will go into that. And we just went and double and checked whether we have more neuroblast-like neurons, and in fact, 2 times. This is double cartin as a marker, specific marker for these neurons. Then we looked at the number of proliferating cells, and by using PCNA, this is marker of proliferating cells, and these are commonly used markers of adult neurogenesis, and in fact, it's increased 2 times. Another two papers, what I would like to say, there was an interesting report in Current Biology recently that some people in Norway, they liked rotten fish. Some indigenous people, they like rotten fish. And interestingly, when they checked TAR5, they have mutation in TAR5. It was a very interesting story. Another story is just because it's related to work in humans, because mice are mice, but what happened in humans? Some German group reported that they can see activation of neurons in amygdala by fMRI after intranasal trimethylamine stimulation. So in humans, we see some responses to some alterations in the function of TAR5 as well, or some function. So the question important now. Now we believe it's a new neurotransmitter system. How individual TARs are involved in this circuitry? What are new natural and synthetic ligands of TARs? Can TARs serve as new targets for the treatment of neuropsychiatric disorders? And that is a major topic of my lab last 5-10 years. What about other TARs? It's pretty much the same story. TAR2 we found also expressed in this in hippocampus. We see projections, how it's going inside of the brain. We see increased adult neurogenesis in TAR2 knockout mice. Pretty much the same story, but what we believe that because they project TARs located in different glomeruli, they will have different projections. For example, from TAR5, it will be more for amygdala, from TAR2 more to hippocampus. And so it will be somewhat close but different profile, not identical. And that's why it could be that potentially by affecting different TARs, we can get different types of drugs. So that's our summary of this work on what we have now. We have studied published TAR5, TAR2, and continuing work TAR6, TAR8, TAR9. So all knockouts we have, and we are working, and we see pretty much similar system. Somewhere more in some knockouts, somewhere less. So that's what I'll my conclusion slide for this part. Interestingly, this story. So the most in frogs, in snakes, in fish, you have huge neurogenesis. In zebrafish, you can cut half of the brain, it will be restored. So, adult neurogenesis is huge there. Being reduced to mammals. In mice or rats, it's much stronger than in humans. In humans, and being reduced to. In fact, there are two groups of scientists. Somebody says that it's quite important adult neurogenesis is human. Somebody says it's practically absent. But majority of people believe that it is, and I belong to this group, that adult neurogenesis in human exists, but it's less. It's less, much less than in mouse. Who doesn't have adult neurogenesis at all is dolphin. Dolphin has no adult neurogenesis. And now I'll tell you a few numbers. The highest number of TARs found in zebrafish, 122. Mice and rats have 14, 17 receptors. Humans have 6. Dolphin has 0. It's not my slide, I just found it. So is it coincidence? I don't think so. I think it's something really, really important, but we have to study, investigate what it means. So, okay, I said it already. So the last slide probably. I should say that recent 10, 15 years, there is a revolution in pharmacology. Many, many drugs now being used intranasally because it immediately goes into the brain, bypassing brain-blood barrier. And there are a lot of even antidepressant recently new appeared one, intranasal administration. So think about that. What if these odorants, these terpenes, what I told you, this cadaverine, putrescine, can enter the brain the same way and activate these receptors not only in our nose, but inside of the brain? Why not? And I checked yesterday, there are a couple of papers about that. That it's technically possible. That's why probably we have now seen these olfactory receptors inside of the brain, because you can potentially affect them the same way like it's demonstrated here by some compounds. So, and just to show you, this very small librarian, we have collection of all tar knockout receptors. Nobody can claim that in the world. Nobody has this collection as we have. And we have in vitro screening platform for tar ligands in cell culture, and it works closely with chemists who synthesize compounds, and we are checking them for activity on one or another tar one. So this is only a short list of people with whom I was working 20 years on this topic, and I'm grateful to many companies who supported us all this time, without whom this work would be impossible. Thank you very much.
Before we go to questions, let me mention that our institute with Dr. Gainetdinov has just begun a study on olfactory influences of these trace amines on social, emotional, risky behavior. And this evening, during poster session, you can be introduced with these studies by our young researchers. Because to our knowledge, not many studies on humans and on this social emotional behavior was done with these trace amines as stimuli.
Okay, so Zarina, if we have some questions online, still we have some. How many minutes we have before the break?
Okay, right now, no questions.
Okay, so we also have the audience. I will ask first question. Raul, thank you very much. Fantastic talk. Since perhaps I would ask whether you can speculate a bit, because many studies of individual decision making, also social interactions, focus on dopamine, and you show that trace amines work closely with the system. Can you speculate perhaps a bit? So what can be a difference of this system compared to the dopaminergic system? So if we would focus on the system that is largely ignored in social neuroscience and neuroeconomics, what kind of function you would assign to this system during decision making or social interaction? I already see, of course, link to odors and social odors, but what else? What is uniqueness of the system compared to dopamine system?
Okay, so we right now mostly investigated TAR1 receptor, and as I mentioned, it's already in clinic, and TAR1 is, as I mentioned, is able to make heterodimer with D2 dopamine receptor. So it's mild modulator of dopaminergic system. So you wouldn't have to work hardly on dopamine. So but in decision making, probably you would have this mild effect. Okay, so it's like I would say modulator of dopamine, serotonin, and glutamate system certainly will make changes in decision making. It's clear. How exactly? Right now it's difficult to say, but I hope that ulataront will appear soon. This first TAR1 agonist in clinic, and you can try in humans and to see probably it will affect seriously decision making. Other receptors, as I mentioned, so-called olfactory TARs, they are certainly would also modulate dopamine system. I just mentioned for TAR5, but we see it with TAR2 too, and with other receptors too. So I believe it will be even more interesting because here you have direct influence of emotional behavior. So you have influence on nucleus accumbens, for example, frontal cortex. So in areas really important for reward for, and it would be, but these things you can test now with these odorants what we have already available, and we are doing it with your group. Yeah, in fact, and let's see. Let's see. I would say let's see what happens. Probably. No, I already can, of course, predict because our principles of aromatherapy already showed alterations in many aspects of cognition, decision making, and so on. And in aromatherapy, used a lot of compositions of terpenes, which I mentioned can be blockers of TARs, many of them. And I'm reading now a lot of about aromatherapy and in fact on emotions, on decision. So it's just, what we should do as a molecular scientist, we should pair certain terpenes with certain receptors. And then we'll see how it will work. Because right now you take, for example, some aroma and you don't know what it is exactly. Ladon, for example. Ladon has huge number of compositions, a lot of terpenes. And I know like 4 or 5% is myrcene, for example, of that. And myrcene blocks STAR5, for example. I know that. But important is now to find, to pair components of this aromatherapy with certain receptors, then take this component and work with that on humans. I mean, on decision making, you can check and see what will happen.
Thank you so
I
much.
hope one, yeah.
And also another a bit naive question. So, I mean, obviously there are the strong effects of trace amines, but I assume that there is a different time scale. Yeah. So, am I correct that with olfactory system you have rather fast effects, but if you consume, let's say, cheese or certain versions of meat, it will be like a longer effect because it should be metabolated and processed and so on and so on. So you would expect different time scales of these effects. It's a bit naive question, but what do you think?
Actually, I was reading yesterday about effect of olfactory administered drugs. I was talking about drugs. They were saying it's excreted like next day. Actually, it's not so short acting. Maybe emotionally it's short acting effect, but if something gets in our brain, it's not quickly being excreted, eliminated there. So I'm talking about regular drugs. About food, normally I guess it's very difficult to get to the brain something what we consume with food because you have to eat probably or drink a lot to get the right concentration in the brain. But in certain conditions, for example, if you use MAO inhibitors, I guess you can get something there. Or you have somebody has mutation, for example, in monoamine oxidase or some other enzymes. I expect that certain part can get into the brain. It's true. But what was described so far with food derived trace amines is only peripheral effects. And as I told you, is cheese effect, so-called cheese effect. But certain part should get it, but not much, not much. Because of blood-brain barrier, and it's difficult to get probably the right concentration of trace amines into the brain through food. But it's not excluded. It's not excluded that certain population can be, and so. Yes.
I have another question. You already mentioned an interesting paper on people who like rotten fish, and I lived 1 year in Sweden. They do like in North Sweden rotten fish. Now you explain me this phenomenon. It was really awful time when all North Swedish, I mean, they smell by rotten herring. Yeah, and they really enjoy it. I mean, I also at some point started to enjoy it.
Rotten herring has highest concentration of trimethylamine.
But also raise as a question. I mean, do we know some people with certain SNPs or certain aberrations of nucleotides that also would allow us to study, like not patient population, but some variations of genes related to trace dopamine? So instead of waiting for some drugs that can block the system, can we do we know about the certain groups and populations that can be invited to the lab and tested because they have?
Oh, actually, I was really having the same question, but a little bit. I'm a mouse psychiatrist. I'm thinking about psychiatry. I would love to see the data of these people with mutation on TARA5 in Norway, if you can get to this population. I really wanted to see the psychological profile. It would be very, very interested because this population is already collected. We don't have it, unfortunately.
Okay, so for example, at least this group is well known and can be addressed. Okay. Other questions?
Excuse me. Yes, we have extra question from Andrei Vlasov. So, Andrei, you can unmute your mic and answer by yourself or ask by yourself, or okay, I will read it. So, could you please, dear Raul, explain in a few words how TARS could potentially be related to COMT or DRDs in the dopamine pathway?
DRD2, you mean? Can I see it? It's in chat, yeah?
Yes, sorry.
Yes.
Yeah, sorry, I switch on. Hello, thank you for your presentation. It's very interesting. So I asking about SNPs like corticolmethyltransferase and other genes related to dopaminergic system, for example, DRG2, free, whatever. Thank you.
I don't think anything known about that. This is interesting question. Yeah, potentially somebody should study. Nothing was studied so far. I haven't seen anything on this topic, but it's very important topic indeed. Yeah, I agree. Please study.
I'm sorry for interrupting. May I ask a question, please? Professor, thank you very much for your speech. It was a great pleasure. My name is Alisa, and I'm one of the executors of our partnership project with ICN on TAR ligands on risk decisions and induced emotions. So you mentioned frankincense and other pioneer woods as a potential source of TAR ligands. And you also mentioned that that some terpenes may be TAR ligands. Does it mean that all terpenoids and terpenes may be potential TAR ligands? Thank you.
Yes, and, the story is terpenes are, as I told you, there are about 30,000 terpenes unknown. I don't think all of them will be ligands, blockers, but many of them should be tested at least. I mean, them monoterpenes, diterpenes, sex terpenes. So there are many of them. And we so far found antagonistic activity to TAR5 with 5 of them, with 3 of them, 3 of them. But we just trying to buy some system and extend this research by find library of terpenes and to do it systematically for with many of them. I expect there will be several of them should be important. The most important, the most interesting for us to find the most potent one and to work them with them. So that's an idea. Terpenes are in all aromatherapy. They use pine woods or whatever. You know, think about that. It's actually there are papers that aromatherapy can increase adult neurogenesis. I found such papers. They just don't know how it happened. What if they found some terpenes which block TAR5 and then it stimulates adult neurogenesis? That can be relationship to Parkinson's, for example, potentially. I mean, it's like I should say, just think about that. If you go to sauna and use eucalyptus, you inhaling huge concentration of terpenes. Just think about that. And it's potentially part of this health promoting effect of sauna as well. Yeah.
Thank you so much.
Thank you very much, Dr. Gainetdinov.
Raul, thank you so much for the incisive talk and for the link of social neuroscience, individual decision-making studies with the newly discovered neurotransmitter system. We're looking forward to use this in practice. And of course, we all have to go to sauna soon. Raul, thank you so much. Now we're approaching our break. Vladimir.
Thank you very much.
Yes, we meet at 14 or 2 o'clock, yes, to listen to other lectures.
And we have break
For 1 hour, yes.
Nina, можно, наверное, на русский перейти. Здесь, наверное, только русский сейчас находится, но язычный.
Мы хотели подарить подарочки участникам, которые пришли вживую, поэтому сначала давайте я вам отдам наши сумки фирменные, а потом подойдем и, да, перекусим.
Алексей, вы с нами?
Я вас слышу хорошо, да. Вы меня слышите, видите? Слышите, пока нет, видите. Меня слышно, коллеги? Меня слышно ли?
Да, отлично слышно и видно.
Все,
Вы
прекрасно.
сейчас ко-хост, можете расшарить экран.
Хорошо, сейчас один момент. Так, видно мой экранчик.
Okay, let
отлично.
me introduce the next presenter, Alexis Belyanin from International Laboratory for Experimental and Biobehavioral Economics, who will speak about interregional trust in Russia, a large online experiment. Please. Алексей.
Алексей, как у нас со связью? Там ничего не зависло, мы хотим проверить, Алексей. Наверное, перезагружается. Okay, so we'll start in a couple of minutes. Alexei is reloading his Zoom. Yeah, we can see him. Alexei, can you hear us?
Can you switch on your mic, please? Yeah.
Yes, да. Слышно меня хорошо?
Yes, perfect.
Okay. There was some issue with Zoom. I don't know what was that. But okay, hope it will not last will not repeat. Okay. Just trying to share my screen. Can you see my screen now?
Yes,
Yeah.
we can.
Okay. Okay. So here I am. Please let me know when I could start.
Please. I have introduced you already, Dr. Belyanin, please.
Okay. Thank you very much. Then it means that I can start. Yeah. Okay. Okay, so thank you very much for the invitation. It was a honor for me to be in this audience, and though I will not be really speaking about neuroscience and about the brain in general, but I hope I will try to convince you that I will be talking something about, maybe let's give you some insights into ecological validity of experimental data, as suggested by the conference title. Okay, so yeah, please interrupt me whenever you have questions, and please let me know when I should stop in about 5 minutes time before that time. Okay, so I'm gonna present results of our long distance real-time economic experiment on trust in Russia, which is actually a joint work of myself and my colleagues from our laboratory of behavioral economics at Higher School of Economics in Moscow. And if you have any kind of suggestions and questions and further ideas what should be done in this direction, I'm more than happy to hear to your comments and suggestions. Okay, so the general question which we address is the problem of trust. As you know, trust is a complex phenomenon. Basically, the OECD says that the key conditions for that are that Persons believe that another person or institution will act consistently with their expectations of positive behavior, which of course says a lot of different things, mentions a lot of different notions, not all of which are straightforward from psychological perspective in general, and of course it's way farther away from explaining where exactly in the brain trust takes place, right? So because the phenomenon is very complex, and in fact, even behavioral data is not exactly straightforward when talking about trust, because as we shall see shortly, right, there are quite some, even though the literature on trust is abundant, I cite only very few papers which basically discuss the role of trust and its implications for social life. But in general, the conventional, what I'm going to ask when I question mostly is the conventional measure, and in fact the conventional conclusion, that the Russians are not very trusting and very trustworthy people. When by trusting we mean how many people say yes on the question on the world value survey, which is on your screens, which is also known as the Rosenberg version of the trust question, which in particular asks, do you think most people can be trusted, or you can't be too cautious when dealing with people? Yeah, so basically this is a binary choice question, and a conventional measure of trust applied to nations is based on world value survey, for instance, or other similar studies, which ask a representative sample of population by countries, a bunch of questions, including this one. And the share of people who say, yes, I think most people can be trusted, is basically viewed as a measure of how trusting is a particular country. Yeah. So as you can see from several statistics here, this is not a very uniform question, which is good, of course, because you expect variance across treatments, and treatments here are countries. And even though Russia, as you see, is 22%, is somewhat below the mean, the global mean, according to the last wave of world value survey, it is higher than the low level. In Zimbabwe, there were only 2%, but much lower than the high level in Denmark and Scandinavian countries, that is typically around 70%. Yeah. There is overall a good correlation, though, of this trust measure with countries' GDP, gross domestic product, right? And as we see, Russia is here, particularly, but other countries align pretty well in this. The correlation coefficient here is probably rather large, something around 0.6 or maybe more. And you see there is quite good correlation of wealth of the country per capita, and agree answers yes to the question on trust. This is not, of course, saying that trust causes high GDP or vice versa. There's endogeneity here clearly, but looking at the picture itself, you can figure out that yes, there is a positive correlation, and we expect countries which trust more, we expect them to be richer. This is not actually quite true, because I don't cite it here, but there is a famous paper published recently by Armin Falk and colleagues from Germany, who have developed a global battery of preference questions, social preference questions, including trust, and they found that things like trust, that is positive reciprocity, willingness to punish for misbehavior, negative reciprocity, rejection, and ultimatum gains, for instance. That trustworthiness, that is willingness to reciprocate for trust. Risk preferences, generosity or altruism, dictator game. So these measures, measured in a variety of ways, be it behavioral experiment or be it survey questions, they are not very well correlated with GDP per countries, and they have covered about 80,000 people and around 40 countries, I think. But the only measure, by the way, which they found to be correlated with wealth of the countries is intertemporal preferences or willingness to wait for larger reward. Patience. Patience is correlated, but the rest is not. And this is, but coming back to trust itself, what is more important is that if you look at this question itself, and can you most people can be trusted or you can be too cautious when dealing with people, it's actually not the only way you can ask this question. And the alternative way, for instance, the OECD version of the question is on a scale from 0 to 10, when 0 is not at all and 10 completely. In general, how much do you trust most people? Right? And this is on another version, which actually shows a higher level of trust than the old value survey version of the question. Further, these further distinctions come from generalized versus institutional trust, that is maybe trust to particular institutions. Like actually, if you think about the general question, and I suppose you are asking this question: how much do you trust other people? And what comes to your mind? There may be a variety of things which may come to your mind. You may think about your neighbors with whom you have just interacted. You may think about your colleagues with whom you have just drunk a cup of coffee in a cafeteria. You can think about your family, about your parents, about your children, about your spouses, about your president, about presidents of the other countries, about the media, about Telegram channels which report various catastrophic or not so catastrophic events globally. So whom do you trust exactly? Yeah. And answering this question, so someone who answers this question may have in mind any combination of all these and a myriad of other possible ideas about what do I mean by most people can be trusted. And actually none of these is observable to the scholar who asks this question. So you don't know what exactly people have in mind when they say so. You measure the aggregate, okay? You measure the aggregate. In a large enough data sample, you may say that you average out all these possible considerations, and you measure general temperature of an average patient in our clinic. Okay, clinic, the whole country is our clinic, right? But then that's fine if you want to compare countries, but it's not fine if you want to draw the implications of trust on behavior of people in the country, because people may have a rather large level of trust, but it may be largely driven by factors which are immaterial for most of economically relevant or ecologically valid actions which citizens in the country do commit, right? For instance, on a real and very much contemporary example, suppose you ask Palestinians, do you trust Hamas? Okay, and the question may have, you may say yes, you may listen yes, or you may listen no, but yes and no, what does it mean? It means that you trust Hamas because it defends you against the aggressors, or it is, so basically Hamas is on your side, or you say yes because you believe Hamas is the only power which is real in our country, and the rest doesn't matter, and the rest cannot defend us against the Israeli bombs. Similarly, no may mean that you disapprove Hamas's actions, or that you would approve them, but you see what kind of disasters they bring on your heads, and as a result you don't trust them. You don't know which of these factors would actually condition the particular answer given to you. So putting aside this extreme example, again, I don't want to speculate on political questions anyway, because especially these ones, I'm not talking about that at the moment. But what I want to highlight is that the meaning of general trust is not clear from that question, from the Rosenberg question. Putting aside as a delicate issue in the first question, another delicate issue in the first formulation, Rosenberg question, you may ask a contrast. Most people can be trusted, or you can be too cautious, too careful. Yeah. So this contrast is explicit in the question formulation. In the old value survey, there is no contrast. Right? Just how much do you trust? Tell me. 0, no, 10, fully. Yeah. 11 possible answers. That's it. So in this formulation, you do not make a contrast. Do not force people to think about the dangers of being trusted. Yeah. And as a result, the results from a paper commissioned by the OECD, which compares self-reported trust on the right-hand side graph, right? Rosenberg and OECD question. And as you see, some several countries like France, Italy, Sweden, South Korea, USA, and Germany, the Rosenberg version leads to systematically lower values of trust in comparison to OECD version. And the OECD question, by the way, is highly correlated with behavioral trust, which is a measure about which we're gonna talk at length. And yes, and there was a recent paper which shows that these two trusts are correlated, but correlation between behavioral trust and self-reported Rosenberg question trust is not very high. And behavioral trust is not highly correlated with Rosenberg question either. So we wanted to, in order to address this question, we come back to, okay, let me skip in the interest of time several more details, but there are some surprising variances in trust, in particular in Russia. As you see, there are large studies covering almost the whole country and discussing self-reported trust across Russian regions. And there are systematic shifts. For instance, in crisis times, 2009, as you see, mean trust is 18%. After crisis and after some other events, in 2014, it goes up almost 2 times, 30%. Yeah. So, but again, if you try to make sense of the trust in particular regions, and there are 500 observations behind each of these bars per region, yeah, I would say it's very hard to do so. At least given the general study, this data comes from GeoRating database collected by FOM in all over Russia. So what do? We do a different thing. We look at a trust from behavioral perspective, so make a behavioral measure, incentivized decision, and incentivized decision in our case is basically the decision about trust to other people in the country. So our laboratory is the whole Russia in this case. And I'm not exaggerating that because I am exaggerating, okay, in a sense that we do not have a representative sample of Russian citizens. That's true. But what we do have a coverage of all regions from Vladivostok here to Kaliningrad here, from Arkhangelsk here to Makhachkala here. And we cover almost all, with a couple of exceptions, almost all millionaire cities in Russia and a few smaller scale cities because of technical conditions, constraints, which I'm going to just discuss shortly. So the our major behavioral measurement is done by the trust game, and in particular investment version of the trust game, which was this version originally was actually due to an economist David Krebs. The investment game version of this game is due to Berg et al. 1995, a very famous paper which is used in many contexts. So there are 2 players in this game. One decides to trust or not to trust. If the first player decides not to trust, then everyone receives 10 units. Right? If the first player decides to trust, then the second player receives the 10 units from the first player, which are multiplied automatically by 3, and then the second player decides to what extent he or she wants to divide it. It is to be honest, for instance, to the first player and share this 30 equally between him or herself and the first player, or grab everything and give the other player nothing. Now we use an standard version of this game. In particular, the first player, both players have 10 units of experimental currency units. In our case, 10 units equal $1. $1 is not our term, it's a term of the crowdsourcing platform, Yandex.Toloka, which we use. And so then if first player passes his or her 10 to the second player, then the second player has a range of possible decisions from 30 to 0, 30 to the first player, 0 to himself or herself, or 0 to oneself, and 30 to the or 0 to the first player and 30 to oneself, with steps of 3 units. So there are altogether 11 decisions here. Now here are the instructions which you use on the screen to the subjects. So there is first player who can keep 10 tokens to itself or share with the other player, which are going to be multiplied by 3 then. If that happens, then the second player can return any share to the of these 30 to the first player. How much would the second player return? So the first player is a truster who trusts or not. The second player is trustee who basically is a trustworthy, so deserves, basically reciprocates to the trust of the first player if the first player gave the money, or is basically a cheater and grabs much the bulk of that to its own pocket. Yeah. So the technique which we use are strategy method. Strategy method means that we do not let players to play this game for real with a particular person, but we do create an imaginary situation for them, and in order to basically get more information from one person than we could do it from physical game. So we say, okay, first players do not know with whom they will interact. So they are somewhere in Russia, in some city, in a Russian city. They can be interacting with any of the 12 cities around in the country, including its own city. So maybe your partner, participant B, will be from your own city, maybe it will be from any other 11 cities which are going to be listed to you on the screen. And you have to make the decision to trust or not to trust. It's a binary decision, pass 10 tokens or not, towards any of these cities, each of these cities. Imagine yourself 12 times that you are interacting with someone from city 1, city 2, city 3, including your city, which one of them, plus the city 12. Do you trust that city or not? Similarly, participants B, players B, have to decide how much they would return to the participant A1 if participant A, or truster, would entrust them 10 tokens, and they would receive 30 by way of this decision of the first player. How much would you return to city 1, city 2, city 3, blah, 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 city 12? Yeah. So this is how exactly our decisions were made. The city names are so far hidden here, are blurred, right? You make a decision 1, yes or no, if you are participant 1, truster. If you are trustee, participant 2, then in the same cities, you make a decision how many tokens would you return to participant 1 out of 12 possible options. So this is, and then we also solicit beliefs or expectations about the other player. So we do it in a way that if participant A, the truster, has to decide has to evaluate that he's told that participant B was told that he or she may receive 30 tokens if you give him or her 10 tokens. So if you were to give your 10 tokens to that participant, how many of the 30 tokens you think participant B will return back to you? Yeah. And they have to evaluate the reciprocity of participant B. And that's rewarded according to the nonlinear scheme. So if you are guessed exactly correctly, then you are received 20 tokens. If you are correct with precision of 1 token, left or right, you are able to use 10 tokens, otherwise you receive nothing. And for participant B, same way, he or she has to make a forecast whether participant 1 from each of the 12 cities will pass 10 tokens or not to participant B. Again, rewarded with 10 tokens for correct answer and 0 for no correct answer. Now, all payments are being made in a crowdsourcing platform, Yandex.Toloka, which is evolving largely in these recent years. For those unfamiliar with that, that's the largest crowdsourcing platform in Russia, and currently it features about 25,000 more or less active participants, of whom about 10,000 are online daytime or central daytime Moscow time, basically, yeah. And so we did it with that platform. We did it online among the participants of Yandex.Toloka. I'm going to give you some ideas where the sample is. It is not representative, as I said before, yeah, but it is still rather large, and it definitely covers people from, not definitely to the extent that the people do not cheat on their location. It covers people from all the regions mentioned in the list, and you can choose which participant from which city will receive which questions. So we use 22 cities for that analysis, for this research, and you'll see the names of the cities. And our goal was to collect 100 participants per row in each city. It basically mostly worked, but for the wave 1, but didn't quite work for the wave 2, which was talking about 1 year after the first wave, in November to 2021. Yeah. So we had with uneven number of participants, but still altogether we have almost 3,000 participants who gave us more than 35,000 decisions. And it was all paid, as I said it before. Yeah, payment was about $4.5 on average plus local fee, and participants had to play that game for us, had to respond to ex-post questionnaire, which was rather large, including actually over 100 questions in the last version, and they had to basically wait till we started the experiment always in real time in all cities, and ended it up whenever we reached the quota of the city participants in each row, or whenever basically there was a natural end that made no sense to wait anymore. There are cities, especially small ones, when turnout rate was not very high. That was one of the main also causes for the strategy facing here, right? So yes, okay, back to results. I think the time is short, so I should switch to results and discuss what did we found. So as you remember, the average level of trust in the country is about self-reported trust. What there is our question is about 25-27%. What do we find in our behavioral measure? We find these statistics. The red one comes from trust, the senders' decisions, share of senders who transferred their 10 tokens to the other player. The other one is responders' reciprocation, so trustworthiness, average share of 30 tokens which they are sending back to the matches. Yeah, the matches. So as we see, there is some variance, but the variance is not huge. On the other hand, there is clear. Not that they're clearly the limited range from about 50 to 67, which is a range of trust in this sample. The bottom ones being, I think, Arkhangels and the Khabarovsk. Yeah, they are about the lowest. But the highest is Makhachkala, following the step. On reciprocation, trustworthiness, it's also rather high. I mean, particularly there's almost never, except for Sochi, maybe below 1/3, which means that if you are getting back more than 1/3 of what the 10 tokens that you have multiplied by 3, you are still getting back something higher than what you would have received had you not been trusting to the other player. Right? So if once you get 40 back, at least this is more than the 10 which you have passed to the second player, so it was worth it. And the average return is 43%, as you see, which means that on average behavioral trust to the unknown person somewhere in Russia does pays off matters. Makes sense to trust this class. Moreover, just to give you a comparison that comparing now behavioral experiment to the experimental data collected globally, you see that there is the first and second players are shown in this picture. They are taken from the meta study of trust in over 20,000 decisions of trust and reciprocation. And as you see, our data marked in red is always kind of above the global trend. Yeah. So they are trusting, and they are more trusting than what you may think. Here are some examples of the cities. Now, coming back to other interesting things besides the fact that we have a rather high trust level. Right? One is that correlation between trust and trustworthiness. How big is that? Well, as we see, not huge, because there are outliers. There are outliers of Sochi and Tomsk, for instance, which have relatively low level of trustworthiness, right, with the average level of trust. And in general, there is a vertical line in the graph corresponding to trustworthiness, basically varies around 0, more or less robustly, right? But trust can be very much shaken over this range, right? From Kaliningrad and Arkhangelsk to Makhachkala and whatever. Yeah. Now there is clear in-group bias in trust. Clear. This picture shows trust to own city and trust to other cities. Trust to own cities is 61%. Trust to other cities is 56%. So the large variance between trust and trustworthiness across the places. Trustworthiness shows same picture. Also, I'm more trustworthy dealing with other cities than with other cities, and that's almost everywhere. There's a big difference between trustworthiness to own and to other cities. Yeah. Validity considerations. Our data shows pretty clearly that, except for some exceptions, like the city of Irkutsk, for instance, on average we have the same level of self-reported trust which you ask in the questionnaire than the world research reports. So this is not an artifact of our data. Is very typical, very similar to the usual pattern of trust preference in self-reported questions, but behavioral trust, as you have seen, is very different. Very different. Here the correlation between the two is not very huge, and in fact the line would be here almost vertical, I think, in general, speaking. Yeah? So this is to say that actually, even despite that there is some more or less fixed and stable level of trust, its meaning for behavior is very much different in different places. Yeah. Okay, let me skip these things and mention the most relevant part of what I'm about to say. That maybe I'll come back to this. No, let me take all that now. Yeah, so basically one of the key questions here from our perspective is what causes high trust. Yeah. Decided that trust is rather higher than they expect. What are the reasons why people trust? And the natural question given by economic theory, of course, these are beliefs of reciprocal behavior. Yeah. So trust game is set up in a way that once you trust, you receive the positive rewards, receive benefits from that trust, and that's why trusting behavior is actually beneficial. People trust because they expect more money, get back more money. Now the question is it really the correct explanation? Can you explain it that way? And in fact, if you think about that beliefs by which you're gonna cross multiply outcomes and make statistically valid decision, right? Should you trust or should you not trust? Is not in fact a very accurate thing, because in Russia, a specific thing, and one of the reasons why we are interested in getting this data from Russia is that there is a variety of attitudes of knowledge of people about other locations. Roughly speaking, we can think about 4 different levels of these connections. One is, okay, you probably know your own city, or city where you have been. Yeah? You probably know capital cities. You know capital cities which are Moscow, St. Petersburg, they are prominent. Yeah? Also prominent cities are in the Caucasus, for instance, Dagestan, or other places in the south of Russia. They are outliers, they are not typical Russian regions. But a third layer is actually a layer of your neighboring regions. You probably have some idea about what your neighboring regions are doing, how people live there, are they trustworthy, maybe have interactions with them, are they trustworthy or not. But so you should have some idea about that. And the rest, the 4th category is the rest of the country. There are many places you probably have no idea what this part of the country is, how people live there, what they are doing for life, how they interact with each other. You don't observe that. You don't know. The country is large, that's why you cannot rely on direct knowledge of everyone about regional specificities of each particular region. Yeah? So basically, if you look at that picture, then there are a variety of things which are interesting, which may be relevant. Namely, is that there are some regions where people are little trusted on the trusting on the others, but there are places when this is not the case. It's the other way around. So to control, to explain why people how people can form the trusting attitude to cities which they do not know, right? We use the following technique, following proxy for that value. We use basically barracks or data about people's residences. About what's people, we use data about people's place of residence when we can infer it directly from them. We said, okay, we live in a particular location. I know about my city, yeah. But what if I don't know about my neighboring city? And we ask that question as a control question. We ask them explicitly, do you have any information about the neighboring city? How people live there? If they do not have this information, right, then how can they really form their beliefs about decisions of people from that location? Our hypothesis is that in this case they use average beliefs about cities neighboring to that unknown city to infer their beliefs about what people in this unknown city are doing. And if you compare their self-reported beliefs about decisions of people in that unknown city to the mean beliefs about cities neighboring to the unknown city, we found that they are very highly correlated. Right? These are very highly correlated measures. So the unknown city is a mean beliefs is in blue. The neighboring known cities are in red. Yeah, there are some exceptions, but they are not common. So basically, people typically do. We have some kind of support with hypothesis that people do form beliefs about these unknown cities on the ground of neighboring cities which they are aware of.
Sorry, 5 minutes are left before questions.
5 minutes, okay, yeah. I'm coming to the end actually, but pretty clearly. So basically, when we have this information, what we can do, we can take a look at what determines people's trust. How much people trust. The trust depends on beliefs, and we use a kind of very similar model to infer these things, and we make this inference on the ground of this simple model of decisions transfer for the senders, which is a binary variable, yeah, depending on the beliefs on a particular destination and a bunch of control variables, and return of the second players again explained by the beliefs and a bunch of control variables. So what do we show? So from here we show that beliefs play different role in different cities. They believe for the senders transfers, beliefs are typically positively affecting their transfer decisions, right? You see in almost all cities except for maybe Moscow, maybe St. Petersburg, maybe Vladivostok, beliefs have a strictly positive impact. Okay? The role of beliefs varies across cities, also to the extent at least, right? For instance, in cities like Rostov or Kazan, it's more or less uniform across the area, right? Or Ekaterinburg. But in Khabarovsk, this is rather scattered, for instance. In Vladivostok, it's also rather scattered. In Makhachkala, it's rather scattered. Yeah? Same way for the second wave. The standard errors here are larger precisely because we do have a small number of observations here, as I said before. And as you see here again, in some places, again, for instance, in Irkutsk, beliefs are always matter. Not in Omsk or in Chelyabinsk, for instance, this is not necessarily always matter, right? In some places, it's rather large. For instance, in Volgograd, it always matters in a very high extent. Yeah. So there is a variety of people's attitudes and differences in their role of beliefs. In some places, people's trust decisions depend on beliefs about the city. In some places, they do not depend on them a lot. Yeah? And this is not necessarily conveying, of course, information about the specific roles of a particular knowledge about a particular region, of a particular person. Trustworthiness, same story, but here the effect is expectedly smaller. But again, you see a variety of patterns. In Voronezh, for instance, trustworthiness doesn't depend at all on the beliefs about whether you receive money or not. In Novosibirsk, it does. It depends, clearly it depends. Again, in Vladivostok, probably in most cases, no, except for Vladivostok and Khabarovsk, own cities, right? In Ekaterinburg, it always does. Does matter. And same stories hold for the other set of cities. So there is a variety of regional differences in attitudes, in the role of beliefs, in the role of attitudes as attitudes towards a particular destination. And yes, we have. I don't have much time to discuss that at the moment, but we have also data showing that this part of these things are consistently explained with the our story about trust, about information about the neighbor, about neighboring cities used as a ground to form your beliefs about city which you do not know and you are not aware of which you are not aware of. Now, other explanatory variables. What kind of social demographic questions or psychological questions do have an impact on your decisions to send or to trust or not to trust? As you see, most there are not many significant variables on the list of variables here. I'm going to spell out those which are interesting. Interesting are you trust more to people who are similar to you. Yeah. In some places, this is a significant coefficient you see. For instance, in Moscow, in St. Petersburg in particular, but in Vladivostok, for some reason, it's basically significantly negative. Yeah. So people trust less to people who are similar to themselves, probably because they have a particular feeling about how trustworthy are their own people. Yeah? And another variable in this variable ready to spend. This variable ready to spend question says, are you ready to spend resources in order to punish those who mistreated you? Yeah. If people are ready to spend resources, they are less trustful. Because, for instance, Khabarovsk, probably because they prefer to substitute these resources for trust. Yeah. So they would rather punish someone who is behaving bad than trying again to trust this person. Yeah? These variables are the most interesting and most telling of all variables here. Same basically techniques and same ideas apply basically to trustworthiness, to you see again trust to more similar strikes in several of these situations right here and here. And again readiness to spending resources also matters for some places. Not much here, for instance, not much here, but there are again some cities when that seems to play, for instance, in Kaliningrad or in Sochi or in Volgograd again. Right. So I skip in the interest of time some of the other details which are available in the presentation, influences and all that. But just to give you some maybe a bit more ideas about how relevant relevant are our data. For instance, trust and trustworthiness seems to not to be very much correlated with religious beliefs. For instance, right? For instance, atheists transfer more than people who have some kind of deviation in beliefs, but trustworthiness is larger for those who are religious. Okay? Big five measures. Again, big five are spread over the regions, and again, the shape of these big five characteristics differs across regions. For instance, in St. Petersburg, the role of extraversion is low, whereas in Khabarovsk it is high, for instance. There are a couple of other considerations which I'm going to mention, but just in the interest of time, let me conclude and let me wrap up a little bit. What do we find? We find that, first of all, beliefs in Russia, behavioral measures, differ a lot from what the World Value Survey reports. World Value Survey tells us, okay, beliefs are low, not necessarily true. If you are given a precise context of trust game and real incentives, people actually are trusting and are trustworthy to an extent which is actually somewhat larger than what the world average implies. There is also a rather large uniformity on this trust level and trustworthiness level across Russian regions. There are, of course, ups and downs. There is no complete uniformity, but still people do have a rather large level of trust to own co-citizens, regardless basically of which part of the country they are in, which basically speaks in favor of the idea that the countries some internal gravity forces which bring the people together besides purely exogenous, say, political will, right? That there are in-city effects which are expected in this case, and there are substantive differences in people's attitude, trust, and trustworthiness depending on whether or not you are aware or you are not aware of that particular city, but people do find an idea how to substitute their lack of awareness about a particular destination by the information they have about some neighboring destination. They can fill the lacunae, which they do not of the lacunae in their knowledge, and by filling these lacunae in their knowledge, they can nonetheless come to a consistent strategy of trust towards other people. Let me stop here, maybe, right, at this point in time, and answer to your questions if you have some of them. Okay, thank you very much.
Let me ask the first question, please. Whether there is a way to understand if all this data could be somehow validated in a real life, like somehow whether we really can give keys to our friends or lend money to our office mates and so on. I mean, real behavioral data. Does someone collect this, or you maybe did this previously?
Okay. Well, there are two layers of answer to this question. One layer would be that this is real data, because this is real money, and people in this crowdsourcing platform, to look, if you know, if you have experience with Zavizat or with Mturk or with Prolific or other similar platforms, people do receive real money for every decision which they make, yeah, which is what we do. So if you are told you have 10 tokens, but you can sacrifice, you can basically risk it in order in hope to get some more money back in reward, then basically yes, you are risking real money. The real question, if I read it correctly, which stands behind what you ask, is okay, but $1 is not a lot. Yeah? After all, you're talking about key from the flat when a bunch of dollars leaves. Can you lend that key to a stranger and hope he or she will not betray you or will not rob you? Yeah? That's a different question, of course, but the different question applies to almost any kind of lab study, which does not go to the field but tries to replicate or reproduce some behavioral patterns in the laboratory environment. So this, if you have some suspicions about the validity of this data for big real decisions, you can address this question to any lab, to any incentivized lab experiment, which also has a modest or moderate, typically a moderate or modest rewards. The question is, how would that be upscaled to real life, real ecological conditions? Well, that's of course a question, but the evidence which exists about this upscaling of lab data to real environments suggests that yes, there are quantitative changes, of course, when incentives are real and big and serious, but they do not kill the effect. So there's going to be still some trust and still some trustworthiness if at stake there's going to be some real fortune, but most people would behave here not as economic theory predicts, of course, not in a rational way. This rational process, full betrayal, no trust. Yeah, but yes, I think what people would do, they would behave in a bounded rational way, or actually demonstrate a kind of satisfying behavior. Once, okay, suppose you trust me for real life and trust me for okay, you gave me $100,000 in hope to get knowing that I will get $1 million. Right? Will I share it fairly with you? Well, probably not. But I will still reciprocate you somehow and give you a good deal of money, something like what you would have expected to get on your own had you not trusted me. So I don't wanna mislead you in a sense that don't wanna make you feel sorry for lending me anything, for giving me the money at all. Right? I'll give you something epsilon over what you would have received had you not trusted me. Right? Would not be completely upset by my misbehavior, but I will not, of course, forget myself. I will keep for myself quite some shares. So in this scale, okay, if one person gives the other one 100,000 and knows that person will raise 1 million, probably this other person will return something like 300,000 or 400,000, keeping the majority for himself, but still sharing in a reasonably fair way.
All right,
so that is
all right. Thank you. Yes, exactly. This I wanted to hear.
Yeah. Any other questions, please?
Perhaps because we're really short in time. Short question, Alexey. Does it look like your data somehow a bit contradicts one of the first slides? At least from the first look, you don't see a link between income of the region and the level of the trust.
Yes, that's true. That's true. But first of all, I will try to look at this picture on income of the region and the trust level. Yes, we did not find good enough correlation on the Russian regions. That's true. So the first slide was on global GDP, GRP of the countries and trust level of the regions. Yes, there is no direct correlation. The correlation is very poor. How it can be explained? Well, I think it's that would I say is as an experimental artifact in a sense, because what we use people from Yandex Toloka, right? And Tolokers are more similar to each other than average residents of a particular region. Tolokers are people who are not only computer literate, people who probably live much of their life next to a computer screen. Yeah. And these people are most likely, like actually our average participant has a higher education, yeah, in all regions, about 48%, I think, of people have higher education. There is a rather larger fraction of people who are unemployed, for instance. There are people who of different professions. Actually, age profile is similar to age profile of Russia altogether. But these things, and there are people living in big cities, which is more important. Big cities in Russia, as you have probably know, about 3/4 of our cities are millionaire cities. If you go to any millionaire city in Russia, you see they are most of them look like downscaled Moscow, basically, right? They have some kind of urbanized city center. They are shopping malls. There are cafes. There are some nightlife, and all these things are very similar. So people who are in our samples, and all these things, they mean nothing. They mean nothing about how poor are people in the countrysides of the respective regions, which are 500 kilometers away from the capital city of the region. Right? So GDP is an average of these countryside people who are also may not necessarily be poor. If you go to you go to Yamal-Nenetsk and Khanty-Mansiysk regions, their per capita GRP is higher than in Moscow. Yeah? So there are wealthy guys in the countryside, but this is not necessarily the case. So I think this is the fact that we do not have good correlation of our trust measures to GRP is due to the composition of our subject pool. Okay. This is of similar people. Yeah.
Unfortunately,
urbanized Russians, higher education, blah blah. Yeah.
Alexey, thank you so much. Unfortunately, we have to move to the next presenter. Alexey, thank you so much. See you later.
Thank you, yes. And the next presenter is our dear friend from China, Zhejiang University, Dr. Fan Shen, who will speak about the art of the deal, deciphering the endowment effect from traders' eyes.
Thank you.
Dr. Fan Shen?
Hi, yeah, I'm here.
Oh, very nice to see you.
So can you see my screen?
Yes. Please proceed.
Oh, okay. May I start now? Okay, thank you. Yeah, I'm very happy to be invited to give this talk here. Actually, I was really planned. I already planned to go to the conference physically, but finally, because some like visa issues, I couldn't be there. But I think it's still good enough for me to give it online. So today I'm going to talk about a recent work I done about how to use eye tracking to decode whether or not a deal can be made during trading. So this work was recently, just last month, or about 2 months ago, published in Science Advances. So it's a still new paper, but my work is mainly about endowment effects. So I think most of the audience are psychology or has psychology or economic backgrounds, so maybe you are familiar with, but at least know endowment and heard about endowment before. So basically, endowment refers to the phenomenon that people often demand much more to give up an object than they would be willing to pay acquirers. So this is originally this phenomenon is originally proposed by Richard Taylor, who won the Nobel Prize in 2017. And later, Daniel Kahneman and Richard Taylor together conducted several classic experiments to demonstrate endowment effects in the lab. So this is a very important phenomenon in behavior economics. So primarily, there are two phenomena for endowment effects. One, what we call is valuation paradigm. That is, in the lab, so the experimenter will give a good, for example, a cup to experiments to the participants. Then participants play the role as a buyer or a seller. Then when they play in the role of buyer, they have to indicate the highest price they will be willing to pay acquire the cup, for example. And when they play in the role of sellers, they have to indicate the lowest they would be willing to accept to sell the cup. So the interesting phenomenon is that they found that the lowest price a person would like to sell a product is often higher than the highest price a person would be willing to buy the products. That is simply because of whether, depending on whether they have the cup like a buyer seller or not have the cup like a buyer, their valuation of the cup will be quite different, indicated by the price. So that is, the willing to accept is larger than willing to pay. So this is a very robust phenomenon in behavioral economics. And then another paradigm, which is often called the exchange paradigm. That is, in this task, participants come to the lab, and then they will be endowed one or two objects. For example, either a cup or a chocolate. Then they can decide whether they would like to exchange what they have for what they do not. So what they found, if the assignment is random, we would expect there are half of people will exchange their what they have. But finally, people find that most people, much fewer than half people, will be like to exchange what they have for what they do not. So this is another phenomenon to indicate endowment. That both of the two experiments indicate that once you have something, you would like to have it rather than change it exchange it for another one. So this is the both, but this violates the basic principles in economics. That is, once there is no transaction costs, so the market should be efficient. People's valuations when objects should be fixed once the ownership is assigned. Regardless, but what endowment indicates that people's valuation for a good depends on whether or not they have it or not. That is, depend on the ownership. So this violates the conventional economic principles and then largely contributed to the emergence of behavioral economics. So there are two classic explanations for the two phenomena of endowment effects. One, what we call is the valuation related explanations. The most famous explanation within this domain is Lossberg. That is, according to this view, that is, Lossberg is another concept in behavioral economics. That is, people weigh loss over gains. So when during cheatings, for the buyer, the good is something they will win from a cheat, but for sellers, a good is something they will lose from a cheating. So if a person is loss averse, then they will of course value the goods more when the person is a seller than when the person is a buyer, according to Lossberg. So this is what I call it valuation related explanation, because sellers and buyers will value their object differently depending on whether or not they perceive it as a gain or as a loss. Another explanation is called what I call it response-related explanations. That is, it means that buyers and sellers have some like response tendencies to prevent them to making the deals. So one of the most popular explanation within this domain is the status quo bias. Status quo bias is that people tend to maintain the status quo rather than change it. So this is also very prevalent biases in our decision making. So according to these views, it well explains the exchange paradigm. That is, if a person tends to maintain the status quo, so what they just to have what they are endowed rather than exchange it for something they don't have because of the status bias. It also can explain the valuation paradigm because for both buyers and sellers, both buyers and sellers have a tendency to maintain the status quo. So if you want to motivate them to make a deal to change the status quo, then you have to incentivize them. So buyers will need a discount in price, and sellers will require premium in price. That will also lead to a gap between willing to pay and willing to accept. So both loss value and the status quo can explain the two paradigms. But of course, we can clearly see that valuation bias, the loss aversion seems to be a relatively better explanation for valuation paradigm, and the status bias seems to be a better explanation for the exchange paradigm. But how to combine the phenomenon? The question is there any way? My question is there any way to like link two phenomena together and the two accounts together? My view is that I would like to unfold this same process. So previously, the research on endowment effects mainly focused on the outcomes, for example, the pricing, or whether or not the exchanges. But I want to know the process, that is, the time taken to make decisions, the information processing reviewed by eye checking, something like this. So I want to use this process approach to combine two phenomena together and to come together. That's my main way. So according to my view, is that that loss aversion should work mainly on information processing, and during this process, people will weigh potential loss over potential gains, and the status quo will mainly work on response selections. During this process, people will have a tendency to maintain status quo and a resistance to change, to make a change. So, but the two paradigms I want also to create combine two paradigms together, create a new paradigm that can be better reflects both tendencies. So first, I will introduce the paradigms we used to demonstrate the phenomenon. So what I call it is called transaction paradigm. It's actually very similar to valuation paradigm. That is, during this paradigm, you will see a lot of different products and a lot of different prices. This is called an offer. For each offer, when someone playing as a buyer, you have to decide whether or not you would like to buy or not. When someone plays the role of seller, he or she has to decide whether or not they want to sell it. So fundamentally, and in my study, I use lottery ticket as a product. The reason I use a lottery ticket is that the lottery ticket has a normative values. For example, in my studies, everyone will have, once they come to the lab, they will have 10 lottery tickets. On the front of each lottery ticket, there's a number. And on the back, there's a gray area. Under the gray area, there is not a number. The number under the gray is a number randomly selected between the front number and the 0. For example, on the back of a lottery ticket with 20 on its front, the number under the gray layers should be a number randomly selected between 0 and 20. So in this case, the expected value of such a lottery ticket is 10, right? So because the selection is random between 0 and 20. So for such a lottery ticket, a participant had to decide whether or not they want to buy it at a specific price. The price can be varies between 1 to 10. And the face value, this is the number on the front of a lottery ticket, ranges from 2 to 20, with 2 yuan as an increment. So every participant, this is within summary design, every participant had to play both the role of a buyer and the role of a seller. And they will play the buyer's roles for a block, making 100 choices, and playing the seller role for a block, making another 100 choices. And fundamentally, their choices actually the quite similar. That is, for when they play a buyer, they are going to decide whether or not they want to buy such a lottery ticket at such a price, and they are choosing buy or not. But fundamentally, they are choosing whether they would keep money or they want to abandon money to get the lottery tickets. And the sellers, when they play a seller, they have to also make a decision whether or not they want to sell such a lottery tickets at such a price. So in this case, they also decide whether they want to choose money, that is to sell the lottery tickets, or to choose lottery ticket, that is to not sell lottery tickets. So fundamentally, buyers and sellers are making the same choices, but because they have different endowments. Buyers have money and sellers have a lottery tickets. So their choices looks different, but fundamentally they are the same. So according to such a task, we can build joint computation models for buyers and sellers. So what do I use is drift diffusion model. So this is a process model in cognitive psychologies, and which assume decision-making and an evidence accumulation process. So in these computation models, the x-axis indicates time, and the y-axis indicates the same value, which are the same tendency and codes. So for buyers, the upper, so these green curves indicate the evidence accumulation process. And once such a process reaches their bottom lines, then buyers will choose not to buy, that is true money, and sellers will choose to sell, that is also true money. So once the evidence accumulation process reaches up lines, up border, buyers will choose to buy, that is true the lottery tickets. And sellers will choose not to sell, that is also true lottery tickets. So for both buyers and sellers, this model can describe their binary choices. And critically, so how to indicate the true biases? First, the status bias will be present on the starting point of the evidence accumulation process. Because both buyers and sellers have a status, we assume both buyers and sellers has the status bias, that is to maintain their status quo. So buyer's starting point will be closer to the choice of not buy, that is to true money. And seller's starting point will be closer to the choice of choosing lottery tickets, that is not sell. So there's a difference between their starting points, that the sellers has a higher starting points than buyers in this case. And another thing is the speed of the evidence accumulation process. So that which it can be indicated by the mean slope of this evidence accumulation process. This mean slope is determined by difference between the face value of the lottery tickets and price. And also critically, we assume that buyers and sellers assign different weights to lottery tickets. That is, seller assign a larger weight to lottery tickets than buyers. So that we will assume WS, the weight seller assigned to lottery, would be larger than the WB, that is weight buyers assigned to lottery tickets. So by doing so, we can incorporate such a response bias, what I call, and the valuation bias together into a computation model. So next, we want to demonstrate this using such a paradigm, as I said, right? So we did an eye-checking study. In this case, you can, and there every time in each block, in the buyer block, the participant will see a price and also the face value of the lottery. For example, then they decide whether they would like to buy such a lottery ticket whose face value is 16 yuan with the price 7 yuan. And also, when they playing as a buyer, sellers, they will decide whether they would like to sell such a lottery tickets at a face value of 16 at a price of 7 yuan. So actually, buyers and sellers see exactly the same set of offers. So there are in total 100 offers in our study. So the y-axis indicates the face value of lottery ticket. There are 10 different lottery tickets. And the x-axis indicates the price. There are 10 different prices. So which results in 10 by 10, 100 different offers. For example, this offer here indicates whether you would like to buy such a face value lottery tickets at such a price, or whether you would like to sell such a face value lottery tickets at such a price. So both buyers and sellers make 100 choices. And so if a person is a rational person, then they should sell all of this, they should accept all of these offers below the diagonal, because for this offer, for all the offers below the diagonal, the face value of a lottery ticket is more than double of the price. So buyers should buy it. Buyers should buy it, but sellers should not sell it, because expected value is half of the face value. And for all of the offers upper the diagonals, above the diagonals, so they have the face value of the lottery tickets is smaller than half of the price. So in this case, buyers should not buy there, and sellers should sell there. So sellers should sell these should accept these offers, and buyers should accept these offers. That is, both buyers and sellers should accept half offers and reject them. And for those offers between right x diagonals, they can either accept it, reject it, and fine. So a rational person should accept half of the offers as a buyer and also accept half of the offers as a seller. So, but what we found is that the result didn't indicate this. That is, for buyers, this is the probability of acceptance. We can say that buyers accepted fewer than half of their offers, and we also see that found that sellers also accepted half of the offers. So both of them are not so inclined to make a trade. And also we can see that response time, that is here response time. We can see that buyers tend to make slower decisions when they decide to buy than decide not to buy. And sellers tend to make also slower decisions when they decide to sell than decide not to sell. That is, both buyers and sellers tend to choose not to do something rather than do something. So they are slower to choose to accept offers, to buy or to sell, than they reject offers. And also we see a main effect of sellings, because that is, people in general are slower to decide whether or not to sell than decide whether or not to buy. So this is probably because most of the participants are students in our universities. So they are not so familiar with selling, but more familiar with buying. Anyway, I think both choice and the response indicate some tendency to not make the trade, to not make a deal. So across different offers, we can also see that buyers accept fewer offers. The red indicates the redder the higher probability of acceptance, the bluer the higher probability of rejection. So we can see buyers accepted fewer than half of offers, and sellers also accepted fewer than half of offers. So this also indicates endowment effects. And response time, the rather the longer response time, we can see that response time is this is slightly below the diagonal, and this is slightly above diagonal. So both across different offers, we can also see some clues for the endowment effects. And we also calculate the difference between what we call relative price. That is the price minus the effective value of lot ticket. That is half of face values. And we can see that when the relative price increase, people tend to sell, buyers tend to sell, and buyers tend to not buy. And the 50% of probability of buying and selling indicate that in different points. We can see that sellers in different points is higher than buyers. That is, sellers values their tickets more than buyer. And also the largest time taken to make the choices also indicates their response in different points. We can see that sellers in different points are also higher than buyers, which also indicates sellers require more monies to give up a lot of tickets than sellers. Than price buyers would like to pay to acquire lottery tickets. So using computational models, we can see that this is using these computational models, we fitted the observed choices and response times to our models, and then we can identify starting points, ZBZS, and the weighting, W and WS. So this is the choices we observed, and this is response time we observed, and this is choices we predicted by our models, and this is response time predicted by our models. We can see that the choices predicted well match the choices observed, and response time we predicted is also correlated with the response time we observed, because response time is very, very noisy, so it's not as well as choices. The prediction is not as well as choices. So according to this model fit, we can identify the starting points for buyers, the red dots, and the sellers, the blue dots, and also weight or lot rates. We can say seller starting points is higher than buyers, and the seller's weight for lot rate is larger than buyers. Then we calculate the difference of starting points between sellers and buyers, and we also calculate the ratio of the lot rate weight between sellers and buyers, and log transform weights. In this case, we formed what we call response bias and what we call valuation bias. So when both of the two larger than 0, which indicates the endowment can be decomposed into a response bias between seller and buyers and a valuation bias between sellers and buyers. But we can also see that in our samples, each dot is the participants. There are many participants fall in the other 3 regions. For example, in this region, people show these participants show the response bias, but not a valuation bias. And for those participants, they show the valuation bias, but not response bias. So we, according to our models framework, there are different profiles of endowment effects. And in conventional study, endowment is seen to be just one-dimensional. In our study, in our framework, endowment can be decomposed into two dimensions. Next, we want to use eye tracking to better illustrate the cognitive mechanisms underlying endowment effects. So why we use eye tracking? Because gaze allocation is highly related to information processing, and response selection is highly related to pupil dilation. So previous studies have shown that gaze allocations reflect and influences that evaluation. For example, when males just vet which of 2 faces are more attractive, they tend to allocate more fixations to the face they judge to be more attractive. And if experimentally manipulates their gaze allocation to the other face, their preference will be changed. So gaze allocation both reflects and influences the evaluation. And in the student literatures, and we also find that when the product is fixated, people tend to value the product more and more likely to buy. And when they pay more attention to prices, they are more likely to choose not to buy. And so in our case, when people decide whether or not to buy or whether or not to sell such a lottery ticket at such a price, they will also look left and right. So if our gaze also influences the valuation, we would expect that when they look at price, the price rate will increase, and when they look at lottery, the lottery rate will increase. So there's such dynamic valuation process in the decision process. So if we can apply such an attentional mechanisms into objective models, we should be able to demonstrate this dynamic process. So this so we have hypothesized that gaze bias should be related to valuation bias. And pupil dilations, pupil dilation has been related to many different mechanisms like surprise, fear, belief updating, and working memories. And recently, it's a conversion view for pupil dilation that it's about mental efforts. People tend to dilate their pupils when their brains work hard, when they allocate more mental efforts. So according to our frameworks, if they have a starting response bias toward their steps pulls, then if buyer tend to not buy and seller tend to not sell, then we would predict that buyers will need to pay more efforts when it should buy than should not buy, and their pupils should be dilated when it should buy than not buy. And also sellers should pay more efforts when it should sell than should not sell. And we would predict that sellers will dilate their pupils when they choose to sell, then choose not to sell. So in this case, we link our endowment effects to valuation bias and response bias, and link them to gate allocation and pupil dilation respectively. So first, let's see where there's such a difference gate allocations between buyers and sellers. First, for buyers, there are very different gate allocation price and loss rates. They like pay what we gains loss ratio is gains the time spent looking at loss rates relative to the total time spent looking at price and loss rates. So we can see that gain loss ratio is very close to 50. That is, they almost paid half attention to price and half attention to the face value of loss rates. For sellers, if you look carefully, you can see that the loss part is slightly brighter. That's the and the statistics indicate that sellers slightly paid more attention to loss rate tickets than the price, and there's a marginal significant difference between buyers and sellers. That is, overall, sellers tend to pay more attention to loss rate or what they have than buyers. And then we calculate different gate allocation to loss rates between sellers and buyers to form what we call total gate bias. And then we found that this total gate bias is related to the valuation bias we estimated with DDM, but not with response bias. Also, next we used attention different models to see whether we can observe such a dynamic process in the evidence accumulation. That is, the weight assigned to loss rate should be larger when loss rate is fixated, and then when the price is fixated. And we do see that, and for buyers, the two red bars, we can see that their lottery weights, the weight of lottery ticket is larger when lottery ticket is fixated than when price is fixated. And for sellers, it's the same. The weight for the lottery ticket is larger when the lottery ticket is fixated than when the price is fixated. And overall, sellers' weight for the lottery tickets is higher, regardless whether the price or the lottery is fixated. And then we can also calculate ratios between the weight of lottery tickets between buyers and sellers. Then like the two bars, and the two and the two. Then we can form 4 different valuation bias, right? So that is the weights of lottery ticket between sellers and buyers. Then we block transform it. We can see that this valuation bias will be the largest when buyers attend to the price and sellers attend to the lottery. That is, when both buyers and sellers attend to what they could lose in a deal. In this case, valuation bias will be the largest. And the valuation bias will be the smallest when buyers attend to lottery and sellers attend to price. That is, when both of them attend to what they can gain from the deal rather than what they could lose in the deal. So we can observe the dynamics of the valuation bias between buyers and sellers during the trading process, depending on their attention allocation or gate allocation. We can also see that we also analyze the first case. Overall, we found that the first case on lottery is the probability of first case on lottery is below 0.5. That is, they tend to more likely, both buyers and sellers tend to look at the price first rather than the lottery ticket first. But anyway, sellers are still more likely to attend to lottery tickets than buyers. There are differences between sellers and buyers. And this first gate bias is also related to valuation bias, but not response bias. And we also make a mediation analysis. We show that the first gate bias effect on valuation bias is mediated by total gate bias. That is, sellers relative to buyers, sellers tend to first look at lottery, and this first gaze bias will be evolved to total gaze bias, which finally transformed to evaluation bias. We also added pupil dilations, and according to our hypothesis, we should observe larger pupil dilations when buyers decide to buy and sellers decide to sell. And we do observe this. We find that the y-axis indicates pupil size. We find that buyers tend to show larger pupil size when they choose to buy than not buy. And sellers tend to show larger pupil size when they choose to sell than not to sell. And we then calculate the difference between their choices to buy and not buy, plus their choice to sell than not to sell. And then we find that this difference is related to the response bias we estimate with DDM, but not valuation bias. And so by doing this, we can decompose the endowment effects into a valuation bias and response bias, and by incorporating the response time, and then by further linking them to gaze allocation and pupil dilation, we found that valuation bias can be selectively indexed by gaze allocation, and gaze allocation difference between sellers and buyers. And response bias can be selectively indexed by pupil dilations. That is, both buyers and sellers tend to dilate their pupils when they choose to trade, then choose not to trade. So this can be a biomarker for a deal, for whether a deal will be made. That is, when both buyers attend to their potential gains, that is, buyers attend to the good, the products, and the sellers attend to the price. And when both buyers and sellers dilate their pupils, a deal is to be made. Our findings can also contribute to explanations for the neural finding about endowment effects. Previously, there's some fMRI studies on the neural correlate endowment effects. Once they found that the ventral stratums checks the difference between that Frank Nielsen work showed that the anterior insular is activated when people chose whether or not to sell. So our two-dimensional framework can provide a well explanation for the two lines of research. That is, the ventral stratum may check the valuation bias, and the insular, which it relates to alertness, alarm, may check the response bias. That is, once we want to make a deal or a choice that can interrupt the status quo, there may be some our anterior insular can be activated to show some alertness. So our funding can also have implications for the marketing or some market designs. For example, if we want to promote deal making, then we should guide buyers to pay more attention to goods, and sellers pay more attention to price. That is, we can guide their attentions to selectively change their valuation. And if we want to manipulate buyers and seller response, then we maybe set some default options or some other settings to change their response tendencies. So our study provides a framework of how to reduce market frictions between buyers and sellers. So this is the work about endowment effects. We decompose it to price and valuation bias. And at a higher levels, response bias is about some priors for our decisions, and valuation bias is indexed by our attention allocations, which is shown in my study. And at even higher levels, the two can be linked elements two-system theory. That's the prior is about our intuition and their attention and about deliberations. So use this framework. I have also previously composed a loss brain. I think I presented work last year, and this work is about endowment effects. And now our team is also working on some other irrational phenomenon, for example, mental accounting, framing effects, disposition effects. So all of them can be decomposed by using the same frameworks. So thank you for listening. If you have some some other thoughts, we can discuss, or maybe we can collaborate in the future. Thank you.
So we have some space for
Hello.
questions. Hello.
Yes, hello. Thanks for a very interesting talk. My question is the following. So, do I understand correctly that it looks like your analysis is based on the assumption that valuation process does not require cognitive effort and therefore cannot lead to the pupil dilation? Because maybe if I'm a buyer, then my decision to buy as a result of valuation process, and that's why it's linked to pupil dilation.
I'm sorry, it's interrupted. I think the connection has. Can you repeat your questions? Maybe
Yes.
with shorter sentences.
Yeah. So am I correct that your analysis?
Otherwise, I think the internet cannot maintain the whole concept.
Okay, so I'll try to be slow. So am I right that your analysis is based on assumption that valuation process does not require cognitive effort and therefore is not linked to pupil dilation? Because it could be that if I'm a buyer and I chose to buy, this is a result of the valuation process, and this can be reflected in the pupil dilation as well.
I still interrupt, but I think I try to combine the difference, like
maybe I can write in the chat.
The words I heard. So you are talking about maybe the pupil dilation can also involve some attentional bias, right? Is this question? So the pupil dilation, what you are arguing is that pupil dilation may not simply reflect the response tendency, but also can reflect the attention bias or information processing. Is it?
So can you hear me now?
I can hear, but not always.
Okay, I can write in the chat.
Cannot the complete. I cannot hear the complete sentence. That's the problem. Yeah.
We will write the question in the chat.
Oh. And I don't know how to like. Oh. And I cannot click Zoom. What's wrong? Yes.
Shafin, can you see the chat?
Here. Sorry. Oh, I can see. Given both stimuli to the same subject bias and seller may be confused. There are studies showing that experience of one role in a game affects one distinct. Oh, it's not your question, right?
Yes, we will type the second question right now.
I'm sorry. Yeah. Yeah. Okay. It seems. There's some issues with. So first question is that yes, I think they will influence it. According in our supplements of the paper, you can see that if people playing the seller role first, we will observe some like slightly larger down effects than if they play their buyer role first. That is probably because seller is a more difficult roles to play. So if you start with a seller role, then you will show larger endowment effects. Okay, the second question is your analysis based on an assumption that evaluation process do not require efforts and hence are not reflected? Oh no, I don't think so. So valuation process is, in our case, I think the effort is more related to the amounts of evidence accumulated. So if you go to the my computation models, you can see that what I the longer distance, the larger distance between the starting points and the border is also related to a large amount of evidence to be accumulated. So my implications for mental efforts is fundamentally related to the amount of evidence accumulated. But you are right. So actually, our attentional allocations should also require efforts. But I still couldn't figure out how to like link these efforts to this to the current efforts I described, like the amount of evidence. So my current view of efforts is more related to the amount of evidence you have to accumulate to trigger a choice. Thank you. But your question is very good. I think I should think about this. Yes, if you allocate a lot of attention, it can also cost efforts.
Unfortunately, we have to move to our workshop. Dr. Feng, thank you so much for your presentation. I hope we will be in contact.
Oh, thank you. Thank you very much.
Thank you so much. Dear colleagues, so we have to move to the next stage of our day. You can attend, Nina, can I just come here for a sec? You can attend the workshop.
So the workshop will be in another building, and I will guide you to how to get there. And there will be a very interesting workshop on voxel and surface-based structural fMRI data analysis. So you're very welcome, everyone, to participate in them. And we meet here again at 6:10. Yes, and yes, short talk, spotlight presentation conducted by students and master students and etc. And they will present their results of the work. I think that everyone will find it very informative, and that's what, yes, visiting them.
Thank you so much, dear colleagues. Thank you so much. So you can also join us online at 10 minutes past 6. Now we have a break for workshop. I will switch.
Good day, everyone. I'm very happy to see you today, and thank you for coming. And I'm very happy to see you today. And today I'm presenting my research under the supervision of Vladimir Kazanogov. Funding for which was provided by HEC University. And let's talk about some background first. And let's talk about some background first. The problem of the link between autonomic nervous system activity and emotions has been studied since the birth of affective science more than a century ago. Since then, several peripheral nervous system measures were shown to more or less consistently predict emotional balance. And here are some of them. So decreased heart rate variability was, in several studies, it was associated with negative emotions, with positive emotions, and increased heart rate increased heart rate variability with with negative emotions. Also, decrease of nose temperature was associated with positive emotions using infrared thermal imaging, and some peripheral measures like startle reflex magnitude and post-auricular reflex were shown to be more high in when participants were higher in case of positive emotions was higher in negative emotions compared to positive emotions. However, recent meta-analysis show that the most of these measures are rather inconsistent, and in our opinion, this may occur because of Good day. Good evening, everyone. Thank you for coming today. I'm very happy to present you my research, which was conducted under the supervision of Vladimir Kasanogov by funding of HSU University. It's called Vegetative Markers of Effective Valence. And let's discuss some background first. So the problem of relation between autonomic nervous system activity and emotions, autonomic nervous system or NS activity and emotions is as much old as the effective science itself is, and there are several peripheral measures that that were shown to consistently predict emotional valence. Some of them are given here. So heart rate variability was decreased in case of net positive emotions and was increased in case of positive emotions. Using facial thermal imaging, infrared thermal imaging, researchers found out that nose temperature was decreased during negative emotions and during negative emotions compared to positive emotions, and some peripheral, not autonomic but peripheral measures such starter reflex magnitude and post-auricular reflex intensity were higher for negative emotions than for positive emotions. But the question is left as recent meta-analysis have shown that no peripheral measure, no ANS measure can be considered as consistent, as there are researchers consistent, but as the limitation of their study, they mentioned the scarce data about the physiological measures during different emotions. So first we need to conduct more studies on this to test this and to more studies to gain more data. Second, maybe it would be effective if we change the approach. So the classical approach implies having some physiological prior to set hypothesis. Okay, prior to set hypothesis, but we chose to. Ничего, я просто репетирую. Нет, ты можешь. Хорошо. Physiological prior, and we've changed this prior, and we thought what would be if we do psychological prior. Or we haven't conducted psychophysiological research. It will be in the future, but for now we have some psychological data. So the problem is, as I mentioned before, is this. So we were interested whether these self-reported sensations in the body correspond to the specific patterns are congruent across participants. If one participant reports easiness in their heart, if one participant feels easiness in their heart when they feel joy, does it mean that another participant will also specific patterns of ANS activation? So our main method for collecting data was structured interview. Here is the list of 10 emotions and 10 body parts that were asked about from our participants. First, at the interview protocol, we have emotion-inducing part. We've asked step by step. We've asked our participant to remember, to recall some memories when they felt joy, tenderness, surprise, sadness, etc. And our participants were 18 to 40-year-old people with no psychological or neurological diseases. That's all about the methods. And we've come to very interesting results. Our main finding is that positive emotions are felt more in the limbs, in the chest, and in the whole body activation. And negative emotions are felt more in the jaw, the throat, and the tears. So positive emotions are felt very distributed comparing to negative emotions. Interesting data was also found when analyzing discrete emotions predicted by bodily sensations. predicted by bodily sensations, and we found that sadness was reported to be was felt more significantly more in the throat and in the chest. For the throat, it was the feeling of lump in the throat, and for the chest, it was the feeling of heaviness in the chest. And joy as well was felt in the chest, and in the whole body activation. For the chest, it was like the feeling of warmness or lightness. For the whole body, it was like the feeling of weightlessness. Um, and for the anger, we found some significant jaw activation, but I'm afraid that it's just emotion regulation artifact. It's not the feature of anger that we feel tension in our jaw, but it's like the attempt to regulate our emotions. And for the guilt, again, the activation in the throat was reported more, again with the same feelings like lump in the throat and dry throat. For the fear, heart, stomach, and limbs activation were reported more. For heart, it was like expressions like heart sinking down, or heart racing. For stomach, it was like the feeling of vacuum in the stomach, and for limbs, it was tremor or temperature decrease in the limbs. For face, for shame, we found significant self-reported activation on the temperature of face. So more participants reported temperature increase on face when they felt shame. So of course there are some limitations of this study. The number of emotion, first of all, number of emotion categories into groups was not equal. In the group of positive emotions, as a result of some data reduction, only one emotion was left, and 6 negative emotions. And the second limitation is that the self-report measures of emotions cannot be considered as fully reliable, especially when it comes to reporting the experience that was a long time ago. But long time ago, long time. In Good day, everyone. I'm very happy to see you. Thank you for coming. Today I'll be presenting my research conducted under the supervision of Vladimir Kazanogov, funded by HET University. It's called Vegetative Markers of Effective Balance. And let's talk about some background first. So the problem of the relation between autonomic nervous system activation, ANS activation, and emotions is as old as the affective sciences. But there are some consistent peripheral measures that were shown to predict emotional balance. Here are some of them. So heart rate variability, decreased heart rate variability was shown to be associated with positive emotions, while it increased with negative emotions. And using infrared thermal imaging, also it was found out that decrease in nose temperature is significantly more than compared in positive emotions when compared to negative emotions. And for some peripheral measures like startle reflex magnitude and post-auricular reflex intensity, it was shown that they are higher in case of negative emotions compared to positive emotions. But there are some questions left, as recent meta-analysis show that none of so there are some examples, but there are a lot of peripheral measures. None of these measures can be considered as fully reliable and predicting emotional balance. So there are some questions. Why so inconsistent? And what else could be the predictors of what else could serve as markers of emotional balance in peripheral nervous system. So approaching the classical approach to this problem was that researchers had some physiological prior to set hypothesis, but we decided to get some psychological prior. So this research is about getting this psychological prior to conduct psychophysiological, more psychophysiological experiments. So our main methods for gaining data was structured interview. Here are 10 emotions and 10 body parts that were included to our interview. So it's important to mention that our interview it contained emotion induction method. We've asked our participants to recall step by step their emotions, some situations when they felt these emotions. And moving to results, our main finding is that positive emotions are felt more in the chest, in the limbs, and the whole body, while negative emotions are felt more in the jaw, in the throat, and in the ears. So seems like positive emotions are felt more distributedly than negative emotions. But analyzing in the domain of discrete emotions, we found more interesting results. So sadness was more often more often felt in throat and in the chest. For throat, it was like the feeling of lump in the throat. For chest, it was the feeling of something heavy in the chest. For the joy, it's chest and the whole body. So the participants reported feeling of warmness in the chest and feeling of weightlessness in the whole body. For the anger, it was jaw activation, but we're afraid that this might be some emotion regulation artifact, but not anger feature. For the guilt, more activation in throat was reported, like lump in the throat or dry throat. And for here, heart, stomach, and limbs activation was reported. For heart, it was like heart sinking down or heart racing. For stomach, it was like feeling of vacuum in the stomach. For limbs, it was the tremor, trembling, or the temperature decrease in the limbs. For shame, face temperature increase was reported more. So of course we have some limitations. First of all, the number of emotions in positive and negative categories was not equal. We had to delete for the reason of that statistics made us delete some emotions. So the final list had only one positive and 5 negative emotions. The second one is that we use the self-report as the measure of emotions. It's not so reliable, especially if you ask about very old experience. That's all. Thank you very much. Good evening, everyone. I'm very happy to see you today, and thank you for coming. Today I'll be presenting my research conducted under the supervision of Vladimir Kosonogov, funded by HSU University, vegetative markers of affective valence. So let's talk about some background first. The problem of the link of the relation between autonomic nervous system activation and emotions is as old as affective science is. So it has more than a century of history, and some since then, several peripheral nervous system measures were shown to more or less consistently predict emotional wellness. And here are some examples. So, for example, increase of heart rate variability was heart rate variability increase was more in negative emotions, but decrease in positive emotions. With using infrared thermal imaging, it was found out that nose temperature is decreased in case of positive emotions compared to negative emotions. And for some peripheral measures like startle, reflex magnitude, and post-auricular reflex intensity, it was shown they were higher for negative emotions compared to positive emotions. But there are some questions left because recent meta-analysis have shown that there is no inconsistent peripheral measure that could be a marker of emotional valence. Of course, they mentioned the limitation of scarce data, so we have to conduct more physiological experiments, psychological experiments to gain this data. And but we haven't done that yet. So we've just changed the approach. So the classical approach implied that they had some physiological prior to that hypothesis about emotions being reflected in some NS measures, but we've decided to ask people themselves what do they feel. So we've tried to get some psychological prior for the further experiments. So our research question is, could subjective state or sensations during specific emotions correspond to the specific pattern of ANS activation? And to gain data, the main method we used was structured interview. Here are the 10 emotions and 10 body parts included to our interview protocol. Important to mention that first, step by step, we've induced using autobiographical recall these 10 emotions, not at the same time, but we induced and asked that way. And our participants were 18 to 40-year-old people without no neurological or psychological diseases. And we found very interesting things that positive emotions are felt more in the chest, in the limbs, and the whole body, while negative emotions are felt more in the jaw, in the throat, and the ears. Positive emotions are felt subjectively very widely distributed along the body, while negative emotions are more concentrated. So there are also some interesting results concerning the discrete emotions domain. We found that sadness was more often felt in the throat and in the chest. For the throat, it was the sensation of lump in the throat. For the chest, it was the feeling of heaviness in the chest. And joy was also felt in the chest and the whole body, but the characteristic of sensation is different. So for the chest, it was like feeling of warmness, and for the whole body, it was the feeling of weightlessness. For the anger, we can see that jaw bar is very high, but we are afraid this could be not anger feature, but emotion regulation artifact. And for the guilt, more activation in the throat was reported. Again, with the same sensation of lump in the throat or the dry throat. And some conclusion some other, and for fear, the activation in heart, stomach, and limbs was more often reported. So for heart, it was like heart racing or heart sinking down. For the stomach, it was the feeling of emptiness or vacuum in the stomach. For the limbs, it was trembling or temperature decrease. And also for shame, facial temperature increase was reported. Of course, we have some limitations. First of all, the number of emotions into categories of positive and negative emotions was not equal. As a result of data analysis, we have to exclude 4 emotions, so we have only 1 positive emotion left and 6 negative emotions. And the second limitation is that the self-report measures cannot be considered as fully reliable methods, especially when it comes to reflecting the experience that was happening a long time ago. Thank you very much. Good evening, everyone. Thank you for coming today. I'm very glad to see you. Today I'll be presenting my research conducted under the supervision of Vladimir Kosonogo, funded by HSU University. Vegetative markers of affective valence. And first, let's talk about some background. So the problem of link or relation between ANS activity, autonomic nervous system activity, and emotions is rather old. It's as old as the affective science itself. So it has more than a century of history. And since then, it was shown that several peripheral markers can be considered as were shown to be more or less consistent markers of emotional balance. Here are some examples. So heart rate variability was shown. It was decreased in positive emotions and increased in negative emotions. And using infrared thermal imaging, it was found out that nose temperature decreases in positive emotions compared to negative emotions. And for startle and for some peripheral measures like startle reflex magnitude and startle reflex magnitude and post-auricular reflex intensity, it was higher. They were higher during negative emotions compared to positive emotions. But recent meta-analysis showed that there is no measure that can be called consistent marker of emotional balance. Okay, they mentioned the limitation that the data is scarce, so we need to conduct more psychophysiological experiments to gain more data, but we didn't do. We just changed the approach. So classic approach was that they rely on some physiological prior, but we aim to get some psychological prior. So what do our participants, so what do people feel when they, in their body, when they experience this or that emotions, and could it be the some prior, some emotions? So as the main method, we used a structured interview. Here are 10 emotions and 10 body parts that were included. It's important to mention that we induced emotions using autobiographical recall method, not at the same time, but step by step. Our participants were 18 to 40-year-old people without no psychological or neurological diseases. And we've got pretty interesting results. And our main finding is that positive emotions are felt more in the lips, in the chest, and in the whole body, while negative emotions are felt more in the ears, in the jaw, and in the throat. So they are like more concentrated than positive emotions. And interesting findings were made in the domain of discrete emotions. So sadness was felt more in the throat and in the chest. For the throat, it was characteristics of the sensation was like lump in the throat. For the chest, it was like feeling of heaviness in the chest. For joy as well, chest and the whole body activation were reported, but with different characteristics. So for the chest, it was like feeling of heaviness, feeling of easiness uh, in the chest, and feeling of weightlessness for the whole body. Okay, for anger, jaw tension was reported more, but we are afraid that this could be emotion regulation artifact, but not the anger. For the guilt, throat activation, especially lump in the throat, was reported more. For fear, heart, stomach, and limbs activation were reported. So for heart, it was expressions like heart sank down, heart raced, and for stomach, it was like feeling of emptiness or vacuum in the stomach. For the limbs, it was like limbs or um, fingers trembling and temperature decrease. In case of fear, and for the shame, temperature increase on the face. Of course, we have some limitations. First of all, the number of emotions were not equal in two categories. We had only one. We had the final set had 6 emotions, as we had to exclude some emotions due to data analysis for some statistics reasons. So we had only one positive and 5 negative emotions. And the second limitation is that self-report methods cannot be considered as fully reliable, especially when it comes to reflecting the experience that happened a long time ago. That's all. Thank you very much.
Elena, hello. Can you hear us?
Yes. Hello.
Yeah, so we're preparing to start in 5 minutes. Meanwhile, you can try to share the screen and the presentation.
Okay. Can you see it?
Yep, just a moment. Yep, yep.
Good.
So 5 minutes ready.
Okay, sure.
Cool.
Good evening, and we have last activity for today. We are going to present our posters from young researchers. We are going to have 6. Yes, all 6 are here. You will have 5 minutes for your presentation, and 5 minutes will be left for discussion, questions, and so on, right? And we begin with Elina Tsigeman from HSU University with the presentation about communication quality in face-to-face and video conferences. Elina, are you here?
Yeah, thank you for presenting me. I want to thank the organizers for creating such a great event for researchers to discuss neuroscience, social neuroscience. And today I want to tell you a bit about our large project which is conducted as a mirror laboratories project with the Krasnoyarsk Medical University. And from HSC side, I'm presenting from the Social and Cognitive Informatics Laboratory in St. P. So our project concerns communication quality in face-to-face and video conferences, and we are conducting quite a large and difficult to create an experimental procedure. I'll briefly present this one. So what we know from studies and from our everyday experience that currently video conferences are used in like every activity that we can do for medical help and psychotherapy delivery, in education, in legislation, for job interviewing, for data collection and research. So all these areas now involve video conferences at some point. However, what we don't know is how this conferencing affects our interpersonal relationship, the contact between individuals, all these variables that social neuroscience deals with. So in this large project, we wanted to compare communication quality in video conferences and face-to-face interactions. And we applied traditional assessment methods like questionnaires, scales, etc. But we also tried to do this via biological markers and digital markers. I'll tell a bit more about them when I will be talking about specific markers and their concept they represent. So here you can see the procedure that we try to go our participants through. So we have planned sample of 60 same-sex pairs of young adult individuals, and a quite important thing is that they must be strangers, and we apply a lot of efforts to make sure that they are complete strangers to each other, because previous research not always indicate whether they are strangers or not, and we know that it affects a lot behavior and outcomes of communication. So here you can see this procedure where we firstly ask people about their individual characteristics like personality traits or social anxiety. And then when they first meet with their partner, they do some collaborative tasks like NASA exercise, survival on the moon, small talk pause. They don't know that this is actually a task, but we know this. We're creating a situation where like, oh, we forgot to do something. You need to wait for 5 minutes. And then we try to see what they do during these 5 minutes while waiting for experimenter to do something. And lastly, they do fictional animal drawing task, which is supposed to be like more or less natural thing of like interpersonal, non-formal communication involving laughter and socializing. And NASA exercise in this case represents more or less collaborative, task-oriented task experience to see how people collaborate on a very specific goal-directed task instead of socializing task. After communication, they report a lot of information regarding their communication experience and satisfaction with the communication. Um, and what is important, why I'm actually on this neuroscience conference, is that we are collecting biological data by asking our participants to provide saliva samples. There are overall 6 saliva samples provided. Like in the very beginning, you can see just a blue dot in the very beginning of procedure, after each task, and then the very end of the procedure. So we want to see how oxytocin level, which is a kind of socializing hormone in people and many other animals. So we want to see whether dynamics of oxytocin reflects attachment and social, whether it can be a social marker in people when they start to get acquainted with a complete stranger.
Now, 1 minute.
Yeah. So here you can see a brief description of all the variables that we are collecting and our predictions regarding this. So I must say that this is a project in progress. So we are hoping to receive something like this. So expected directions here. And what I also want to say is that we are very happy to collaborate on other researchers on this large project because we are not sure that we can digest this huge amount of data. So if you have any ideas how this data can be digested, we're happy to invite you to our team. Thank you.
Thank you so much. So some questions. Okay, perhaps I can ask a question. So you mentioned that you have 30 hours of recording. So the overall duration of, am I correct, collaboration will be around 30 hours?
I'm sorry, I'm not sure that I've got the last sentence.
I think in the previous, if you go to the slide, one slide back, yeah, below you have more than 30 hours of audio video recording.
Yeah.
So it means you will record 30 hours of collaboration, or what does this 30 hours consist of?
Yeah, thank you for this question. We are recording collaboration process. It's like 40 minutes approximately for each pair. So we're going to try to process this in terms of speech content analysis, speech production, and many linguistic variables will be assessed as well, like distance between semantic categories in communication, which might be a marker of mimicry or like social synchrony. But we also want to analyze emotion and like emotional expression of individuals to see whether, for example, positive emotions present more in face-to-face or in video conferencing, whether people show similar emotions, because we know this can like transmitting the emotions to the interlocutors to see whether they are similar. So yeah.
Elena, yes, I see the point. I mean, I have a practical question. So let's say when you will record faces in let's say in Zoom, yeah, most probably, I don't know how would you arrange it, but it can be that you have a much higher quality of video recording. How would you record videos in face-to-face communications?
Yeah, we are having several cameras directed to participants. We record the Zoom during the video conferencing, but we're also having a couple of cameras directing to participants from the side in both conditions. So we collect like several points
okay,
for
so
the same pair.
It's really complex.
Yeah.
Another question is, I think you mentioned oxytocin. Am I correct?
Yeah.
So I missed it a bit. Perhaps I just missed the point. Would you measure somehow oxytocin? Or?
Yeah, so we are collecting saliva samples and we are going to extract oxytocin level using immunoassay analysis. This is not the thing I'm good at because we are having colleagues from medical university in Krasnoyarsk who are going to conduct this analysis.
I just wonder, do you collect it, let's say, a few times, so like measuring baseline, or I mean, if you have 30 hours of communication, would you have regular samples, or I mean, it sounds very complicated. I mean, even it's very interesting and exciting. Yeah.
Yeah, thank you. So yeah, we are having 6 saliva samples for each participant in this procedure. So you can see blue small dots here, numbered with these Roman numbers, and this indicates saliva samples which participants provide during the
Okay.
procedure.
Yeah. I see. Now it's clear. Thanks so much.
Yeah.
I think we are out of time. Yeah, so we have to move to the next slide.
No, I have one more question. How are you going to assess theory of mind?
So we are having interesting procedures that we collected from previous research. So what we do is we are asking participants to provide their big five. And at the end of the procedure, after they collaborated with this another partner, they try to provide big five estimates for their partner. So it's kind of
Someone already has done it in literature.
Yeah.
It's supposed to be interesting, yes.
Yeah, it's kind of a measure of social sensitivity whether you manage to read your partner's big five traits.
All right, interesting. Thank you.
Yeah, thank you.
More questions? No.
I think we have to proceed to
Okay.
the next one. Thank you so much.
Thank
Good
you.
luck.
And the next one is Alexandra Morozova from HEC, who will speak about neurophysiological mechanisms of effective misinformation correction.
Thank you. As it was said, my project is on neurophysiological correction of misinformation. And why is it important? As you probably know, misinformation is a huge problem because it spreads fast. It influences people's way of thinking. And gladly for us, there are known psychological traits which which influence the perception of different misinformation, namely the fake news. Fake news, I'm sure, have faced them. Fake news are fabricated information that looks like real news, but was made with not the same intent, and their process of creation is not the same as in real news. And the psychological traits responsible for their perception are different cognitive failures, emotions, person's worldview, social cues, and others. And there are special strategies to counter fake news, pre-bunking and debunking. The first one is when we prevent the perception of fake news before the fake news appears, and debunking when we correct the information presented in the fake after the fake appears. And current study is focused on the correcting news from the medical field using debunking in the form of disclaimer. And its goal to investigate using an EEG technique whether the correction via this disclaimer is effective. EEG technique which is used in this study is event-related potentials, namely feedback-related negativity. Event-related potential is the changes in EEG voltage, which reflects the preparation or execution of some mental or motor process. And the feedback-related negativity is a special ERP component which elicits from 200 to 300 milliseconds after the feedback, some loss or negative outcome. It looks like this in the graphs. And my methodology is following. We have 80 80 news headlines, 40 fake and 40 real on different medical subtopics. And we have disclaimers, which looks like this blue stamp right here. The disclaimers will be true and false and from authoritative and non-authoritative source. The authoritative will be the World Health Organization, and the other one will be International Association of Alternative and Traditional Medicine. And we will also use cognitive tests to measure people's cognitive abilities because they influence fake perception. And as I said, we will use EEG, 64 electrodes with current characteristics. And the procedure of the experiment will be the following. First, we will present the headline, which will look the same as the Russian news in social services or different newspapers on the internet, and then we will assess the trust rate and willingness to share their news on the internet or with others. And then we will put a disclaimer on the news, and then we will assess the rates all over again after the disclaimer. And we have three types of hypotheses. We have a behavioral one that trust rate and sharing rate will significantly change after the true and false disclaimers from the authoritative source. The psychophysiological hypothesis is that the false disclaimer will elicit a stronger FRN compared to the true disclaimer, and this amplitude of FRN will correlate with the magnitude of person's opinion change. And we also expect that people with higher cognitive test results will evaluate the veracity of the news more accurately during the first presentation of the headline. Sorry, and we had a pretest on 15 participants, and our results are following. We found out that trust rates significantly changed after almost all the disclaimers, except the false non-authoritative disclaimers. And this means that our behavioral hypothesis is working. But for share rates, there were no significant differences. And after conducting an ANOVA test, we found out that trust change indexes are significantly different between some types of disclaimers. On the picture, you can see that the difference is present between false authoritative and non-authoritative, true authoritative and non-authoritative, and authoritative or true or false disclaimers. That is all. And next week, we are starting EEG procedure. That's the references. Thank you.
Questions to Alexandra?
Yeah, so do you control in some way for the emotional perception of the headlines? Because I guess they can be positive or negative, like vaccination is good or vaccination is bad, or if you take these vitamins, you're going to be healthy, or if you don't take these vitamins, you're going to die or something like that.
We do not control the emotional reaction, and as I know, the information in the headlines is kind of neutral. It does not say anything about death or being more healthy or less healthy using that type of treatment. It's generally neutral, but suppose for some people the content of the headline could elicit some emotions, but I suppose it may influence the results, but it's I suppose, the different study how emotions influence the perception of the information. It's a good question because using FRN in response to correction, it's a kind of new technique. We are the first who are doing this. So probably we will face some issues, but it's hard to predict what issues and will the emotions affect the amplitude or not. But I think we will find it out soon.
I would suppose that it will not affect, but you could use facial myography, for example, as a control variable. But supposedly it will not affect so much. Yes, of course. Exactly. Please, who wanted?
You provide the truth sign, right? Who is the provider of this sign of truth?
Excuse me, sorry, can you speak using microphone because we cannot hear anything?
I repeat once again, so we have 4 types of disclaimers. So each true or false sign could be accompanied with authoritative or non-authoritative source, but it's not the official position of the organization. We designed it like this, but it's not what the organization are thinking about the news, actually. That answers your question, or okay, continue then.
Okay, but do you think it's real changes, or it's like trustworthy to particular organization on the second route of your experiment? Will be.
I suppose our news are already fact-checked, and I suppose some correction would correspond to the real position of the organization. I am 100% sure that World Health Organization thinks the same about vaccines. They It's hard to say if they will really change it, or whether they will react as they change it. But we hope they do change their opinion. They do change their trust rates. So maybe their opinion is changed, or they wanted to be seen as people whose opinion is changed. There is a thing of continued influence effect that those news can influence them and their perception of reality, perception of thinking, even after the correction, despite their position being changed or not. So it's hard to tell. We need, I think, a longitude experiment with a second test procedure to measure whether their opinion really changed or not. But it will happen in the future, I suppose.
Thank you, Alexandra. And we go further. Lilia Faizova, HC University, presenting the influence of interface elements on trust in online news.
Yeah, okay. Good evening, dear colleagues. I'm glad to present my topic, which is named the influence of interface elements on trust in online news. Where should I press? Ooh. Okay. So I will introduce with this beautiful Goldilocks metaphor, which says that there are a lot of online news, as it was discussed today, and a lot of them are rumors or fake or some weird news or something. And I just wanted to know whether the interface influence somehow the perception of online news. So do the elements of online news interface somehow influence how do we perceive them, whether we perceive them credible or not they're credible. And according to the literature review, there are several groups of elements that influence trust. And among them, there are surface elements, message elements, user-based elements, and social or reputational elements. And in this study, I just focus on surface elements. And I'm just talking about the appearance of the site, appearance of the online news, and the organization of the interface elements. And the methodology of the study can be divided into 3 stages. In the first stage, I do a small pretest on online news just to know whether people read on which platform do they read online news, do they use some verification practices in perceiving online news, and so on. And then the conjoined analysis is in progress right now. I will tell about this in details a bit later. And then an eye tracking experiment, which is also in progress. And I will not stop on the first stage for too long. It was just a questionnaire designed, which was spread on students, and it consists of 153 respondents. And interesting results here is that there is a high level of transition to the source side. So people receive online news in some social media, and then they usually go to the original site when the news somehow interests them to check whether it's credible or not. And as for the conjoined stage now, conjoined stage of the research, it is aimed to identify the influence of different interface elements on perceived credibility of online news. So um, conjoined is a method which comes from marketing studies, and it means we took some levels, some attributes, and each of them has some levels. So in this case, I have 5 um, attributes, each with 2 levels. So overall, I have 32 stimuli. So 32 combinations of all these elements. And the stimuli look like this. So in this slide, you can see 2 interfaces with different combinations of different elements. And the conjoint questionnaire looks like this. So I'm asking a question, um, and the respondent needs to choose which interface do they trust more, or they trust each similarly. And then they perceive this question several times. In my case, it's 14. 14 times each respondent perceives through this question. And yeah, I heard your question about what's the difference between those interfaces. And that's a good question because sometimes they look really similar and the elements are really like really small. And in this case, you can see that this extra field is present here, not present here. And author is here. Oh, they are pretty similar in this case. And I can show you, yeah, for example, this one. The results of the pilot study says that there is a winner, the variant which was chosen the majority of times by respondents, and the one who was not really oftenly chosen. And interestingly for me, winner does not have an author, and the last one has an author. Uh, that seems controversial to some other literature review results. And also menu here is present and here is hidden, and yeah, this is it. Um, Thanks. And as for the last one, this is an eye tracking study. Based on the previous conjoined stage, I will define the good, like the good interface, and the bad one. And I want to put a true news and a fake news into both of those interfaces. So I'll get 2x2 experimental design with a successful and unsuccessful interface layout and a true and false news message. And I want to know what the fake and true news are perceived differently in different interfaces. And yeah, I'll just skip it. And yeah, I just wanted to note again that this is a study in progress, and I'm still collecting the data for the conjoined study. And I will apply regression and clustering methods to make results. And yeah, this is it. Thanks for attention.
Questions?
Okay.
Can I ask a question? So, I mean, can you say a few words about your selection of criteria? Yeah, because what exactly you manipulated? Because and what was the rationality behind it? Okay.
Yeah, thanks. The choice of elements is was a long process. First of all, I analyzed the literature, which was analyzed there, which elements do all the researchers analyze, and then I looked at real Russian news sources based on their top 10, and I looked at what do they have in common and chose those elements which are present in like half of them. Like I didn't choose, for example, the search, which is present in every interface which I found, or I didn't use, for example, a profile, like the icon of a profile, because it's also almost everywhere. And I also excluded some social elements like comments and likes and shares because I think it's another stimuli which makes some social interaction. I just chose those, and I was limited by 5 maximum elements due to the analysis. And yeah.
Final small question. I mean, when I look to your different versions of interest, by the way, it's very interesting project. Yeah. For example, some of the effects, I don't know, Vladimir perhaps would disagree or agree with me. Some of the effects can be driven by attention. So you put the name, and it's not the name per se, yeah, but you diverge attention from certain content. You can have counterintuitive results. Yeah, it can be counterintuitive result. Yeah, so you basically just divert attention from the message. That's why you, for example, trust less. So how would you control for this?
That's a good question, and I think I haven't thought about this. And yeah, that's a good comment about. I just wanted to show you the look. This is a empty slide, and this is a whole slide, and it's really like a lot of attention would be put onto other details here, and here it will be put on the text. And I don't know, the stimuli. I mean, the text and the picture are similar in all the conditions, but yeah.
Yes, a very small question. You presented the same text a lot of times to the
Yes.
same subject,
Yes.
but will it be interesting for the same subject to watch a lot
The
of times to the
point,
same?
the point of this is to measure how people react on difference in the interface. I'm not trying to evaluate how do they analyze the text or the picture. And I think that after a person would be shown this picture several times, they will just get used to the text and to the
Exactly.
picture, and they'll see on the details of the interface.
Maybe an idea would be to show different text to different subjects,
Yeah,
enlarging
we
highly
discussed it with
the
my
sample.
yeah, we discussed it, but we decided to put on this variant because in this way we will not like catch different attitudes towards
So
text.
how many times the same text was presented to one subject?
14 times.
14. 14. All right.
14.
All right.
Yeah.
More questions.
Maybe we have one. Okay.
Am I right that you used also politics news? Politic news. There was a slide with politics something. No? Okay, then the question is absent.
So I just have a menu which has a topic named politics. Yeah, here. But the topic, the text which is presented here is like health connected. And it was in the first connected to COVID, but then we decided to change it to like the influence of chocolate on blood vessels or something. A story, yeah. Thank you.
Please, Alisa Godovanec from our institute will present neuropharmacological effects of TAR receptor agonists and antagonists on neurocognitive mechanisms.
So thank you, Vladimir, for presenting me. Firstly, I have to say that it's a big project that is conducted with my colleagues, Sofia Ponomaryova, Anna Shestakova, Ksenia Panidi, Vladimir Kosanogov, and also in close cooperation with Professor Raul Ganidinov, whose lecture you may have heard today. So let me make a quick recall on TARS. So trace means associated receptors are a class of G protein coupled receptors, and they were discovered in 2001 by 2 independent research groups. Actually, TAR1 receptor is known for its connections with our internal trace means such as phenylethylamine, tyramine, tryptamine, and also there are some ligands in synthetic psychoactive substances that makes it an interesting near pharmacological target. But the receptors from TAR2 to TAR9 are known um, as olfactory receptors in humans and animals, and we in our research mainly focus on them, and especially on TAR5. There is some evidence that TAR5 modulates specific domains of cognition. as it involved in emotional behavior, even adult neurogenesis, and as well in some pathways regulate dopamine and serotonin transmission. Uh, there is a lack of research on humans. The only behavioral research was made on putrescine. It's a TAR5 agonist. And as it was shown in the series of behavioral experiments, um, the exposure to putrescine increased vigilance, caused avoidant behavior in humans, elicited some implicit cognitions related to escape and threat, and even increased hostility toward an out-group member in behavioral experiments. So this what makes TAR receptors such an important topic in means of social behavior. And why orders? Simply, the answer is that we're not a medical college and we cannot use ligands that people have to ingest. And the easiest way to study the ligands is to use some olfactory stimulation. And also, it is very convenient that, unlike other sensory modalities, olfaction appears to have a more direct connection with brain regions associated with emotional responses, as it maintains direct links with the limbic system. So here you can see potential pathways of TAR receptors' influence on emotional reactions and risky behavior. So let me tell you about the stimuli that we have chosen. So we have three classes of odorants. The first ones are agonists of TAR receptors. The first one is trimethylamine, as Professor Raul Ganidinov mentioned today. It's a metabolite of choline and with characteristic smell of rotten fish. The other one is cadaverine, is metabolite of another amino acid, L-lysine, and has odor of putrefying flesh. Sometimes it's even called the smell of death. So cadaverine is agonist of several TARs, as known for today. We also have antagonists, timberol. It's a synthetic molecule that has woody ambery smell. It's pyrophoric antagonist. We have a lot of evidence on this molecule. And frankincense. Some components of it were also investigated by Professor Raul Ganidinov. And also to control whether the influence of agonists and antagonists of TAR is elicited, we wanted to take some aversive and pleasant smells that are not TAR ligands. So we decided to take citral. It's pleasant citrus scent and caproic acid that is very fatty, cheesy, and very aversive for a lot of people, like a negative control. So our main hypothesis is that we expect to see statistically significant differences in means of risk economic decision making and induced emotions under the influence of TAR receptor agonists and antagonists, comparing to pleasant and unpleasant control odorants. Moving to methods. We plan to conduct a series of behavioral experiments. So to assess the propensity to make risky decisions, we will use lottery games. So you can see here on the screen an example of a gambling task using the MPL method. So the thing is that the participant have to choose higher reward with lower probability to win, or vice versa, lower reward with higher probability to win. And to assess the delay discounting, we use binary monetary choice task using MPL model. So basically, the participant have to choose whether he wants to receive more money in the delayed future or less money in the nearest future. And in means of induced emotions, so we try to assess the changes in response time of emotional attribution, also changes in recall time of visual stimuli, and the accuracy of how participants uh, um, recall the visual stimuli. And all the pictures for experiment on induced emotions are taken from a special base called Emma Madrid. They all, so there are three categories: the neutral pictures, the positive pictures, and the negative pictures. There are 270 pictures overall for experiment, 90 for each category, and of course they all presented in randomized contrabalanced order. So Okay, yeah, I'll be quick. So the sample, we will have a quite big sample of 96 participants, 24 participants for each combination of the odorants. And each participant will come 5 times to be presented to each odorant. It's like 480 observation in total. And the procedure is the following. First, we collect data on participants that may influence their olfactory perception, anosmia, parosmia, smoking, allergies, and all the stuff that can influence the olfactory perception. Then we make a physiology test on sniffing sticks to assess level of olfactory acuity using special tables. Then the participants assess the intensity of a particular odorant from 0 to 9. Then he fulfills the tasks, and then again assess the intensity, the familiarity, and the valence of the odorant. We decided to make it in the end, not to influence the answers of the participants, not to focus the attention of the familiarity on the valence. So we also made a pretesting. So here you can see a trend on intensity lowering of odorants. They're quite similar. And on the valence, as it was supposed, frankincense appears to be the most pleasant, and trimethylamine supposed to be the the least pleasant. In further research, after conducting the behavioral experiments, we plan to use FNRS with some potential targets in prefrontal cortex, especially ventromedial prefrontal cortex, orbitofrontal cortex, and maybe DLBFC also, as these regions are known to be involved in decision making under risk and processing social and economical information. So thank you for your attention.
And as far as I checked, it's related to the third class of toxicity. So I just say you should get the possibility to conduct this experiment from bioethic committee.
Thank you for your question. Actually, all of the TAR5 agonists are in some way toxic, but it depends on concentration and the time of exposure. Actually, we asked this question for Professor Raul Ganidinov, who's a neuropharmacologist, and the concentration that we use is non-toxic, and the whole time of presentation for one participant is 1 hour maximum. It's like more 30 minutes. There will be no toxic effects, and even to make our participants safer, we make 2-3 days pause between the trials.
Next question.
Sure. I'm sorry.
Two questions. So first one, I missed a bit. What is your hypothesis related to risk taking? So would it increase, decrease, or you would expect opposite effects for two types of odors? In which direction it will drive risk taking? Perhaps I missed it.
So we expect that TAR ligands answers under TAR ligands influence will be different from the control odors. But the second, the second hypothesis based on other behavioral evidence that the agonists, like aversive TAR5 smells, they increase risky behavior. And so we suppose that, firstly, TAR agonists and antagonists would differ from control, and in means of agonists and antagonists, aversive will increase risk taking, and pleasant will decrease it.
It's also related to Vladimir's expertise. Could it be a confounding factors? Let's say negative averse stimulation would move person into different risk taking versus more pleasant stimulation. So it's not about TAR, but it's about emotions.
But that is why Alisa will have control.
But how does it work with the control? I mean, I would just say it's not about TAR, and not about the smell of cadaver. It's about negative emotions.
Am I understood you correctly that you asked about the emotional pictures? Yeah. Yeah, so exactly that's why we took an aversive smell that is not a tar ligand, an aversive smell that is tar ligand, and we'll compare it between. So
Citric and another one is carpoil. What, the pleasant not trace amine is citric.
Citral and
Citral
caproic acid.
and caproic
Citral
is
pleasant and caproic acid is
they are not
unpleasant.
trace amine.
Yeah. Sure.
The last question.
It's not. Hello? It's clarification. You only use this stimuli as a order, as a TARS. You're not collecting the TARS as a biomarker from subjects, right? Okay, thanks.
Thank
you. Let me invite Firuza Komilova, who will speak about vegetative markers of affective valence.
Thank you. Okay, I was advised to concentrate not on details but on ideas. That's why my slides are very informal. I guess it's not a problem for you. Okay, I will presenting our research under the supervision of Vladimir Kosanogov and funded by HSU University, vegetative markers of affective valence. Нажала. Okay, let's talk about some background first. First of all, why is it important? Why do we measure emotions? It can be nowadays can be useful to study, first of all, consumer behavior in counseling, psychology, etc. And why do we need autonomic measures? Because as we know, central measures are more precise. Because autonomic measures they are less time and money consuming. Talking about the background of this question, it is as old as the affective science itself is, and there are some measures, ANS measures, that were shown to more or less consistently predict emotional valence. So increased heart rate variability was associated with negative emotions, while decreased with positive emotions. And using facial infrared thermal imaging, it was found out that nose temperature decreases in positive emotions and increases in negative emotions. For the peripheral measures such as startle reflex, magnitude, and post-auricular reflex intensity, it was shown that they are higher when in negative emotions comparing to positive emotions. But recent meta-analysis showed that these measures also cannot be considered as consistent markers of emotional valence. And we ask why. First of all, they emphasize such limitation as scarce data. So more psychophysiological experiments should be conducted in order to have more data and do better meta-analysis. But we've decided to change the approach to make another approach. So before, researchers used physiological prior to set hypothesis. So we try to make a procedure which will allow us to get not physiological but psychological prior. So our question is, could the subjective sensations, bodily sensations during specific emotions correspond to the specific pattern of autonomic nervous system activation in our subjects? So our methods, the main data collecting method was structured interview. Here are the 10 emotions and 10 body parts that was included to the interview protocol. First, we induced emotions using autobiographical recall method, step by step for each emotion, and then asked about their sensations, participant sensations during these emotions. We had 45 healthy subjects aged from 18 to 40. And we got pretty interesting results. Our main finding is that positive emotions are felt more in the limbs, in the whole body, and in the chest, while negative emotions are felt more in the jaw, throat, and ears. More interesting results were drawn from analyzing discrete emotions. So sadness was felt more in the throat, and the characteristic of this sensation is like lump in the throat. For the chest, it was the feeling of heaviness in the chest, and the joy also is felt more in the chest and in the whole body. But the characteristic is different. So the sensation in the chest during the joy is the feeling of warmness or weightlessness for the whole body. And the activation sensations in jaw, the sensation of tension in jaw was reported, but we're afraid that this is not anger property, but just emotion regulation artifact. And about the guilt, it was more felt in the throat by the same sensation like lump in the throat or dry throat. Fear was most often felt in heart, stomach, and limbs. For heart, it was the feeling of heart sinking down or heart racing. For stomach, like a feeling of emptiness or feeling of vacuum in the stomach. For limbs, the feeling of trembling or feeling of increased temperature in the limbs. And for the shame, increased face temperature was reported most often. As of course, we have some limitations. First of all, the number of emotions in two categories, in positive and negative emotion categories, was not equal. Because of some statistical reasons, we had to reduce emotions. So we had, for analyzing, we had only one negative, or one positive, and 5 negative emotions. And the second limitation, of course, is that self-report measures cannot be considered as fully reliable, especially when we ask about our participants about the experience that happened a long time ago. That's all. Thank you.
Couple of questions. So first, does it mean, can you come back to one of your results? Let's say, yeah. I mean, you consider the joy as artifact here. Yeah, sorry.
Thank you for the question. Our participants mentioned the feeling of tension in their joy when they experience anger. But as the interview was like open, I hadn't had a chance to ask them questions. Most of the time, I asked them, "Do you feel like it's like you are regulating your emotions, or you feel anger like this?" So most of them answered that maybe it's some emotional regulation, but of course, no one knows for sure.
I have a related question, or can be related to the next one. I mean, I remember that for one of the emotions, you have like limbs involved. So my question actually is relatively simple. If I will manipulate somehow, let's say, the sensitivity of your jaw, let's say, person will chew chewing gum, or I would do something with limbs. I have no idea. sport activity and so on. Would it be predictive for change of emotions in this respect? That's a question number one. And the second question, still, perhaps you can explain how different it is, this approach from the literature you presented, let's say on thermal information, also Ira Yaskilainen made some of the studies. How different is your project from the previous one? Yeah, the first is whether you can manipulate emotions in this, yeah, William James kind of fashion. Yeah, or what do you think?
Thank you very much for your questions. Actually, my own opinion is that we cannot. We just try to find out some non-direct measures of emotions, but it doesn't mean that I don't really agree with this James idea. So I think that no, we can't manipulate and induce different emotions. As for the second question, um, yes. So they based on the previous literature describing emotions like uh, oh there must be they just set hypotheses not based on the subject's own information about their bodily sensations. So they didn't ask where do you feel. Yes, yes. But we haven't conducted psychophysiological experiment yet. It's just our maybe prior or these results should serve to set more solid hypotheses in future psychophysiological experiments.
Yes, thank you. Now we have the last speaker, Sangram Becerra, who will present emotional word valence evaluation and cortical excitability TMS study.
Good evening, everyone. So this is a very interesting topic which we did recently. How do we evaluate emotional words? A big question. To get into this topic, we did some background. We came to know that okay, sorry. So we know that there are mirror neurons, not just visual mirror neurons, but auditory mirror neurons. And these neurons facilitate to experience empathy, to experience different states of emotions, to assess creativity, and apart from that, to analyze others' behavior. We also came to know that from ERP studies that valence is experienced first before arousal. And sometimes emotions are also perceived a little bit earlier, like around 370 milliseconds. Based on these ideas, we went ahead by trying to figure out when we hear words, will there be any effect on the cortex or not? And if there are any effects, what kind of effects? And does the emotional component of words have a say in that or not? We had 37 participants. We had some dropouts due to data exclusions. And we used single pulse TMS to take MEPs from the FDI muscle. For words, we used the INRUN database. We selected 132 words, equal categories of neutral, positive, and negative. They were screened for frequency, arousal, and the number of syllables. And we used free software Balabolka to convert the text to words. We used a feminine voice because the male version sounded more robotic, and we wanted it to be more naturalistic. This is a very simple layout for what we did. First, we collected a few baselines at resting state. Then we presented a neutral video for 2 minutes, 34 seconds. Then the second set of baseline were collected. Then we went ahead with the auditory verb presentation with simultaneous stimulation, which happened just after the finish of the word, or a 500 millisecond after the word presentation. And then finally, a third set of baselines were collected. So to understand what is 0 millisecond and 500 millisecond, when the TMS was applied just at the end of the word presentation, we took it, for us, it was 0 millisecond. And when there was a delay after the word presentation of 500 millisecond and TMS pulse was applied, for us, it was like a label of 500 millisecond. And you can see that the duration of the sounds lasted around like 0.76 seconds. Results were quite exciting. As compared to 0 milliseconds, we found a huge decrease in the MEV values, which are significant for 0 and 500. Valence per se didn't have any statistical impact, but we started seeing a downtrend. As you can see, for neutral, there is not much of a difference. But when it came to positive and negatives, we saw a huge difference. So to conclude why there was a cortical inhibition, we have no idea. We have an inhibition, but all we can do is speculation. Because this was something new and innovative, which we tried. Nobody had done this using TMS, especially to probe cortical excitability with words and emotions. We suggest that participants were still processing the words to understand maybe meaning processing is happening, to understand the effect of valence, whether the trend is real or it is just a variability. We are still collecting some more participants, and we also suggest using more neuroimaging tools like EEG in combination of TMS or fMRI to understand, the whole picture of it. And thank you so much. Thank you for the lovely team for making it a success. And I'm open for questions.
Questions.
It's a lovely project. Can you come back to the setup of the experiment protocol? Yeah, next one. Yeah, just a clarification question. So, am I correct that person was exposed to words as a kind of stream of words, or as a text? I missed it a bit. Or this is during this auditory words with valence. So it's a stream of text of words. Is it a coherent text? Is it a story or a single words? How was it presented?
Single words, nouns.
Have a related question. So would it be a good control study when you would, for example, because you have an interpretation, yeah, that people still process words, yeah? Would it be a good control study if you would, for example, induce emotions using like emotional videos and so on, yeah? So you would, I mean, ideally you would induce the same balance of emotions, but without words, you would expect less cognitive load and whether you will have a different effect. What do you think?
I did go through some of the papers that used videos, and we also got the understanding that for even if it is for videos or words, at N400 there is already semantic understanding, so people know it. And then there were so many contradictory TMS studies, like if you hear negative, if you observe negative valence stimuli, and they were kind of, they had taken the mirror aspect into it, and they found that for negative, the cortical excitability is way off charts. But for us, it is like something new, and we are still trying to understand what is happening and why it is happening. So
I'll give you in a second. So, am I correct? A major counterintuitive result is that their MAP are inhibited, yeah? Because I would expect that people should be more alerted and they would have to be more sensitive and ready for action. Yeah. So in a way, that's the most counterintuitive result. That's actually it's inhibited. Am I correct? And also, there is the next question.
I have a comment for this, but I have a questions first. Okay, how you control the volumes of the auditory stimuli, and how you control the different levels of perception of auditory stimuli?
To control the volume, we found a literature review that when you present words at neutral prosody, which doesn't have the tone, so that somebody doesn't know what kind of emotion they are kind of listening, we used manual control of volume. And the paper suggested that there is no effect of volume on emotion perception. So we did had our check. Individual subjectivity, that thing we still haven't controlled it.
I have a comment for this too, but I have another question. How you study without control, without with words, without any meanings? There are several studies. Maybe it's not include the TMS, but they study also words, auditory, and they use words without meaning. For example, bar, and etc. No, without meaning. Words without meaning.
That part we haven't checked it, but it's a good suggestion.
For questions?
Really exciting project. I just wonder if you can do. I mean, now it's pure speculation because I'm also puzzled by your result. Yeah. For example, I mean, what comes to my mind, like negative emotions, or let's say related to stress, yeah, related to like freezing and action. So at the same time, you can have both in a way, yeah. And it depends on even on the muscle you use. I mean, would your results change if, for example, you would use an active paradigm and person could, let's say, because you most probably measured MAPs for fingers, so yeah. So, for example, if you would use fingers for certain reaction, let's say, to escape a um, painful electric stimulation, yeah. So in this case, this you kind of you're ready to for action and do something. Yeah? And if you would change MAP in this case, that would be a bit more behaviorally valid because the whole system is preparing for a certain action. And you would see, let's say, that negative emotion potentiate, for example, yeah, your response and so on. I just wonder, I mean, it's a pure speculation. If you can introduce like a behavioral response related to this muscle, that would more or less make sense with if emotions would change reactivation of this region. Wild idea, perhaps very stupid, but yeah.
I mean, I concur to your idea. Like this study was we the idea came out of pure curiosity. It is an exploratory project, and the results what we have derived and taking your suggestion into account, it opens up a new scope and can be diversified for many other future studies. I mean, we take words for granted when we speak to ourselves and to other people, but this, I mean, a layman even can understand that words have effect in our brain. So if we are more sensible enough, the implications are huge.
Just maybe one last question. So am I right that you're looking at the mirror neuron reaction? So like, for example, when you show this and you like look at the response to this gesture, and is it different from just simply motor cortex excitability in general? Because maybe the trick is that it's not just motor cortex excitability, but it's response to mirrored action.
That's the reason we did check about mirror neurons and especially auditory mirror neurons and their interplay with empathy and modulating emotions. In this paradigm, we didn't check mirror neurons. So that opens up another whole idea of expanding this experiment. Just you listen to words, they are relaxed. We just tell them to pay attention. We don't even tell there are negative, positive, or neutral words. They don't even know what to expect, words or phrases.
Thank you, Sangram.
Thank you so much.
Yes, we have finished for today, and tomorrow we continue at 11 o'clock. We will have 4 lectures. We will have the continuation of our MRI workshop, and we will have another evening poster session. So you are all welcome.
Recording stopped.